Human Health and Disease, The Lessons from Genomics, organized by Professor Partha Majumdar. And we have a galaxy of eminent experts in Professor Mark Stoneking, Professor Mark Daly, Professor Arindam Maitra, Andreas Rice, and Partha Majumdar himself. I must tell that it took me 2 years to be able to get Partha Majumdar to do this symposium. And it has already taken me 10 minutes to even bring him online. But before we request him to talk about the symposium, may I request Professor Chandrima Saha to introduce him formally, and then he can take over the symposium. So, Professor Chandrima Saha.
Thank you, Professor Singhvi. I welcome all fellows who are present here and the distinguished speakers to this session. Can you hear me?
Yes.
Okay, so it's really a pleasure to introduce Partha Majumdar, who is known as the Gene Guru in the media. He is currently the president of the Indian Academy of Sciences, Bangalore, and a national science chair at the National Institute of Biomedical Genomics at Kalyani. He was the founding director of the same institute, NIBMG. Professor Majumdar is internationally acclaimed for his contribution in the field of genetics and statistics. His key interest has been the human evolution, and he has worked assiduously to uncover genetic architecture behind disease susceptibility and drug response. Professor Majumdar has been honored with several medals, including the Trust Biology Prize, the G.D. Birla Award for Scientific Research, Ranbaxy Research Award, Om Prakash Bhasi Award in Biotechnology, New Millennium Science Gold Medal of Government of India, and J.C. Bose National Fellowship. He is a fellow of all the 3 academies of India and the World Academy of Sciences. He has served on the board of directors of International Genomic Epidemiology Society and was the founding chair of its ethics committee. He also worked on the International Bioethics Committee for UNESCO. He has been a passionate science communicator and frequently communicating through various media, simplifying and communicating science to the public. Professor Majumdar was elected to INSA Fellowship in 2001. I welcome Professor Majumdar to take over and conduct this very interesting symposium, which I'm sure is going to be a feast. Professor Majumdar.
Thank you, Chandrima. Thank you very much for this very kind introduction. And I also take this opportunity to thank Professor Ashok Singhvi for calling upon me to organize this symposium. It's really a pleasure. First of all, I owe an apology to all of you, primarily because you're unable to see me and it took me a long time to get on the system. I don't know what has happened. I'm unable to turn my camera on. Be that as it may, it's really the 4 people who are actually the major players today or major drivers of this symposium today. All of them good friends of mine, Mark Stoneking, whom I have known for many years. I'm not going to formally introduce Mark Stoneking. Mark Stoneking was actually the person who found this out of Africa, or who proposed this out of Africa with a lot of scientific evidence out of Africa theory of human evolution. It was a theory at that time, but the evidence that he provided and that has accumulated over a long period of time is actually now a fact that humankind evolved in Africa, came out of Africa to populate other parts of the world. He is going to talk about archaic genomes today, and as we might remember that the title of the symposium is Human Health and Disease. Human health and disease has a lot of genetic underpinnings, and these underpinnings have a continuity in evolution, and that's the reason why I thought that it would be a good idea for Mark to provide us with an overview of how we have evolved, such that the 2 speakers who will follow, Mark Daly and Arindam Maitra, who will speak to us on genomic underpinnings of diseases, can put them in perspective of human evolution. So without further ado, let me also state that Professor Mark Stoneking is a professor at the in Leipzig in Germany, and he's also been elected a fellow of the U.S. National Academy of Sciences. And we've known each other for many years, and he is really a wonderful speaker and has done a lot of interesting evolutionary genomics work. Without further ado, Mark, all yours.
I may only mention that if you have any questions, kindly type it out in the chat box, and then Professor Partha Majumdar or some one of us will read it out, and then the concerned speaker can respond to it. Thank you.
Also, let's all mute our microphones.
Right. Well, I hope everyone can hear me, and I will start the screen sharing. So
We can hear you very clearly.
good. Okay. And let's see here. Okay. So thank you very much for that introduction. I hope people can see my slides. And yes, I've known Partha for many years, and I'm very happy to participate in this symposium. Unfortunately, virtually rather than being able to be in India for real, but we have to cope with these circumstances as best we can. So what I want to tell you about today is insights into human evolution that come from archaic genomes. So when people think about research in human evolution, the images that usually come to mind are the paleontologists and archaeologists who go off to exotic locations and dig up evidence of our past, usually in the form of fossil remains and also in the form of our cultural remains like stone tools, pottery, and so forth. But beginning in the last few decades, there's been another source of information about human evolution that's become increasingly important, and that's studies of this molecule of DNA. And how is it that we can learn about human evolution by studying variation in DNA? Well, it's quite simple. Even though it's not so exotic, perhaps the locations where we do this work. Occasionally, we do get to go to interesting places to do our field work, our sampling. And the reason why we can use DNA to study human evolution, of course, is that DNA is inherited from our ancestors. So we got our DNA from our parents, who in turn got their DNA from their parents, who in turn got their DNA from their parents, and so forth. So, and this process goes on for tens of thousands, hundreds of thousands, even millions of years. And so there's a record in the DNA of the people living today of their evolutionary past. So we can get insights into human evolution by studying variation in the DNA of people living today. And what's even more exciting, and has been made possible in the last decade or so, is by studying DNA retrieved directly from fossil remains. Okay, so I want to start with discussing this particular question, and I'm going to take a sort of historical perspective here, probably because I'm getting rather old myself, and I tend to think that history is important. So how did our own species, anatomically modern humans, originate? Now, when I began working on this question as a graduate student back in the early 1980s, there were basically three hypotheses that were being discussed as to how humans originated. So one of these shown here is called the multi-regional evolution model. And the way the evidence for this goes, and this is all fossil evidence now, is that prior to about 2 million years ago, all of our evolution is taking place in Africa. So there's no good evidence yet that anything older than about 2 million years ever made it out of Africa. But beginning around 2 million years ago, something gets out of Africa, and people argue about what it should be called. Here it's called Homo erectus, and spreads across the old world and was quite successful. And according to multi-regional evolution, most of the evolution that occurs in these different parts of the old world is indeed descent from the archaic populations in that part of the old world. But there's also gene flow going on. So wherever mutations arise that are important for becoming modern humans, they get spread by gene flow to the rest of the old world. So according to multi-regional evolution, you have the entire old world population of archaic humans. So spread from sub-Saharan Africa to Western Europe to Eastern Asia, even out into Oceania, evolving in concert to become us, to become anatomically modern humans. Now, in contrast to multi-regional evolution, which would say there is no single origin of our species, There are other theories that propose that, yes, our species originated in a specific place at a specific time. And the most prominent of these back in the 1980s was the out of Africa hypothesis, which again was proposed first on the basis of fossil evidence. And according to the out of Africa hypothesis, everything happens in Africa until about 2 million years ago. Beginning around 2 million years ago, some of our ancestors get out of Africa. They spread across the old world. They are successful for hundreds of thousands of years. But then between around 200 to 300,000 years ago, something special happens in Africa. And that something special is us. And we become modern humans. We start spreading across Africa. And ultimately, between 50 and 100,000 years ago, we leave Africa and we start spreading across the rest of the old world. And in the most extreme version of the out of Africa hypothesis, which is out of Africa with replacement, the modern humans coming out of Africa don't interbreed with the archaic humans that were outside of Africa, like Neanderthals, for example. Instead, they completely replace them without any interbreeding. But then there is a third variety of hypothesis, and there's actually a number of these, but I'll just lump them under the term assimilation, which combines out of Africa, but with some contribution of archaic non-African lineages. So according to assimilation hypotheses, modern humans do arise as a species in Africa 200 to 300,000 years ago. They spread out of Africa 50 to 100,000 years ago. But when they meet up with Neanderthals and other archaic humans, they don't completely replace them. Rather, they interbreed with them. And so they pick up some of their ancestry as well. And there's different assimilation hypotheses that propose different amounts of this interbreeding, different places and different times, but they all have the key feature that they propose that there should be some archaic non-African ancestry found in modern human populations today. Okay, now I want to emphasize that these three hypotheses were all first proposed based solely on fossil and archaeological evidence, not on any genetic evidence. And yet it proved incredibly difficult to distinguish between them using only fossil and archaeological evidence. And I think that's because the problem with the fossil evidence is that we don't have, living, breathing Neanderthals and other archaic humans to compare ourselves to. Instead, we only have bits and pieces of their fossils, and those have to be put together and reconstructed. And it's from those reconstructions that we then make our interpretations about how similar or how different they were from us, how they behaved, were they behaved the same as us or different from us or not. And in this process of doing these reconstructions, you get biases creeping in. Okay, and here's just one example of what I mean. So here's an artistic reconstruction of a rather complete Neanderthal fossil. It's the old man of La Chapelle. And you see, according to this reconstruction, that the Neanderthals were very fierce and hairy and brutish. And this guy's carrying a big club, and he's probably going to whack in the skull of some poor modern human coming around the corner here. And, you look at that sort of reconstruction, you say, well, if that's what Neanderthals were like, maybe they were your ancestors, but they weren't my ancestors. And now here's another artistic reconstruction of the exact same fossils. And now you see something that looks very modern, right? He looks very much like us today. And he's making a stone tool, and he's obviously thinking very hard about what he's doing. And he's wearing a necklace, which Neanderthals didn't really have, but, never mind about that. You look at this sort of reconstruction, and you say, yeah, if that's what Neanderthals were like, then I have no problem with thinking that they were my ancestors. So I think these caricatures, and they really are caricatures, no one today would think either of these was a good representation of Neanderthals, but they make the point that what these sorts of reconstructions tell you about is what the people who made them thought about Neanderthals. So how can we get beyond this issue and actually try to distinguish among these fossils, these hypotheses? Well, what I and others argued was that even though these three hypotheses were proposed based on fossil and archaeological evidence, they can be distinguished by what they predict about genetic variation in modern humans. Because the way they can be distinguished is what they propose in terms of the contribution of African ancestry in non-African populations. And we can put this on a continuum. So at this one end here, we would have a recent African origin with complete replacement, which says that all of us get all of our ancestry, 100%, from the South Africa event, right? Down towards this end, we would have multi-regional evolution, which would say, yes, some of the mutations important in becoming modern human arose in Africa and spread out of Africa, but others arose in Europe, others arose in Asia, and they get quickly spread around by gene flow, so we lose the signal where they would have originated. So we don't really expect to see a strong signal of African ancestry in non-African populations. But then in between these two, further toward this end, are what I call the assimilation hypotheses, which would say that yes, the majority of our ancestry comes from this out of Africa event, this African origin and out of Africa event. But some detectable amount of that ancestry, and you can argue about how much it should be, 5%, 2%, 1%. Some detectable amount of our ancestry should be coming from these non-African archaic human populations. So this view would say if we want to distinguish among these hypotheses, let's look at the genetic data. And the first gene to be looked at in sufficient detail to test these hypotheses was not from our nuclear genome, but it came from a different genome, the mitochondrial DNA, which is located in the mitochondria outside the nucleus of the cells. It has a number of nice properties. Perhaps the most important is that it's transmitted from mothers to sons and daughters. So we all have mitochondrial DNA, but only women pass it on to their offspring. Therefore, we get insights directly into the maternal history of populations. And a study that I was able to participate in with Rebecca Kahn and Alan Wilson, we analyzed mitochondrial DNA variation from human populations from around the world. And to not go through the details, but just to tell you the essential features of what we found that there was by far the greatest variation in mitochondrial DNA in African populations. And that's a signal of an African source. Moreover, the diversity out of Africa is a subset of the diversity in Africa. So strong indication of an African origin. We see that when we do phylogenetic trees, they indicate an African source for modern human diversity. And we can use a molecular clock approach, the idea that mutations occur at a regular rate through time, to ask how much time would it take to produce all of the mitochondrial DNA diversity we see in modern populations today? And the answer is around 200,000 years ago or so. So all of this implies a recent African origin of human mitochondrial DNA. But we know that the history of one gene, one locus, like mitochondrial DNA, can differ from that of the species as a whole because of chance events or maybe because there's been some selection operating. So we need to look at other genes if we want to see if there was really a strong signal of a recent African origin across our genomes. So the next gene to be looked at in sufficient detail to do this sort of work is this poor, pathetic, puny little chromosome here. This is the human Y chromosome. It's what makes us males male compared to an X chromosome. And phylogenetic trees of human Y chromosome variation look very much like the mitochondrial DNA story. We see the deepest branchings in Africa, the most diversity in African populations, and it dates to around 200 to 250,000 years ago. So the Y chromosome shows a strong signal of a recent African origin. And of course, we are now in the era of genome-wide data. We can look at variation across all of the chromosomes. We can do this with SNP arrays, or we can do it by whole genome sequencing. And we construct trees based on these sort of data, and they also show a strong signal of an African origin. And with genome-wide data, we can do a lot more than we can do with just mitochondrial DNA and the Y chromosome. For example, we can do computer simulations. So we can simulate demographic histories in the computer and then compare the patterns of variation that are produced by these simulations to what we see in real populations today, and ask what sorts of demographic histories give us the best match to the patterns that we see today. And those always involve a recent African origin. So there is an overwhelming signal all across our genome. It's not just mitochondrial DNA. It's not just the Y chromosome. It's all across our genome of this recent African origin. Okay, and so that's sort of now the textbook view. So it's easy to find these sorts of diagrams with all these arrows and nice dates going to show the origin and spread of humans. But keep in mind that when they were spreading out of Africa and even across Africa, they're meeting up with all of these archaic human populations. And so did they interbreed? So in other words, is it out of Africa with replacement or out of Africa with some assimilation? Or in other words, what people, that's how scientists talk about it, but what people really want to know is, did modern humans have sex with Neanderthals or other archaic humans as they spread out of Africa and across Africa? And it proved incredibly difficult to answer this question using just modern genetic variation in modern human populations, because there'd be studies which say, oh, here's a pattern that looks unusual at this gene. It must be something that came from Neanderthals. And that would be followed a few months later by another study saying, well, no, you can actually explain that with the out of Africa with replacement. So it went back and forth, no clear resolution until we were finally able to get genetic information, genome-wide data directly from fossils. So Neanderthal genomics is what finally provided us the answer. And the first Neanderthal genome sequence is published now 10 years ago. It was a low quality sequence. It's a draft sequence. It has lots of errors, but it's still sufficient to address these questions. It was a composite sequence that came from these 3 rib bones that all came from this cave, and this is Vindija cave in Croatia. These remains are all dated to be at least 38,000 years old. So now in this study, there were 3 different statistical tests that were done to ask the question, do we see Neanderthal-related ancestry in modern human populations today? All 3 gave the same answer, so I'll only talk about one of them. And I'll skip the gory mathematical details, but I do want to go through the logic a bit, just so you can appreciate how one goes about making these sorts of determinations. So one of the predictions we would make is that if there was admixture between Neanderthals and modern humans, the Neanderthal sequence should match alleles variants in some humans significantly more often than in others, and those that didn't receive ancestry from Neanderthals. So here's the test that was done. That was to compare 2 humans with different alleles, one of which is ancestral, the other of which is derived, for which the Neanderthal has a derived allele. Okay, so what do I mean by ancestral and derived? Well, this is what the actual data would look like. We have a sequence from human 1. Here's our DNA sequence. Here we have a polymorphism. Okay, so here we have a difference between these 2 humans. So at some point, an A mutated to a G or a G mutated to an A. So how do we know which is ancestral and which is derived? That's fairly straightforward. We compare this to chimpanzee and other non-human primate sequences. So if now we add in a chimpanzee sequence and we see that it carries the A at this position, that tells us that A is ancestral, G is the derived allele, direction of mutation was from an ancestral A to a derived G. Okay, now, if we go through and look now at sequences from 2 humans and the Neanderthal, most of the time what we see is that the gene tree, that is the tree for the gene sequences, matches the species tree. So the species tree is the expected relationships, which are if we compare a Neanderthal to 2 random humans, let's call them Donald and George, then what we find is at the time both George and Donald will have the derived allele and the Neanderthal will have the ancestral allele. And that will reflect mutations that occurred from the ancestral to the derived state on this branch of the phylogeny. So that's all fine. Or we'll find that both Donald and George carry the ancestral allele and the Neanderthal carries the derived allele. And those would be mutations that occurred on this part of the tree. And that's all fine. And when we compare 2 human sequences to Neanderthal, the vast majority of the polymorphisms that we find will fall into one of these 2 cases. But there will also be some exceptions. Okay, there will be cases where there's a mismatch between the genetic sequences and the species tree. Those are cases where the Neanderthal has a derived allele, one of the humans has a derived allele also, but the other human has the ancestral allele. Now, it turns out there's 2 ways we can explain these mismatches. One of them is if by ancestral polymorphism. Okay, so if the mutation from the A to the D, from the ancestral to the derived allele, occurred in a population that was ancestral to Neanderthals and modern humans, so that we have both A and D alleles, the ancestral and the derived alleles, present in this ancestral population, then it can happen that just by chance, the Neanderthals get the derived allele, one human gets the derived allele, and the other human gets the ancestral allele. Okay? The other possible explanation is that there was gene flow from Neanderthals into modern humans, so that there's a mutation that occurred from ancestral to derived on the Neanderthal lineage, but then subsequent gene flow from the Neanderthal into the ancestors of Donald, but not the ancestors of George, bring the derived alleles there. How can we distinguish between these 2 possibilities? It's pretty straightforward, because here we expect the Neanderthal to match the 2 humans with equal frequency. If it's ancestral polymorphism, whether Donald gets the ancestral allele and George gets the derived allele, or the other way around, doesn't matter. It's like a coin flip. Okay, so here we expect the Neanderthal to match the 2 humans with equal frequency. Here we would expect the Neanderthal to Donald more often than George. So that's the basis of the test. We take diverse human genome sequences from around the world. We compare them to the Neanderthal and ask, do we see any excess matching? And if we take 2 sub-Saharan Africans, we see no excess matching. We take 2 non-Africans, here in this case, a French individual and a Papuan from the highlands of New Guinea, we see no excess matching. But where things get interesting is if we compare a non-African to an African, then we always see a significant excess of matching to the non-African. And it doesn't matter if it's a French individual like shown here, or someone from South Asia, someone from East Asia, someone from New Guinea, someone from the Americas, they always show an excess of matching compared to Africans. Okay, so this sort of summarizes it. Compared to Africans, no excess matching. Compared to non-Africans, no excess matching. African to a non-African, we always see an excess matching with the non-African. And so the way we would explain that is that when modern humans left Africa, we don't know where they left, but this was one possible route. Wherever they left, they quickly met up with Neanderthals, they interbred with them, they picked up some Neanderthal ancestry, and then after the interbreeding with Neanderthals is when they started migrating into the rest of the old world and diverging. And that's how you get essentially the same amount of Neanderthal ancestry all across the old world, and ultimately even into the new world, is by migrations after the interbreeding occurred. So in other words, okay, so we come back to our models for modern human origins, and we can convincingly rule out multi-regional evolution and out of Africa with replacement. Instead, what we have is assimilation. Or in other words, yes, modern humans did indeed have sex with Neanderthals. Now, in the course of doing this work, Svante Pääbo, the head of our department, was trying to get his hands on any potential Neanderthal fossil he could find to see if it had enough DNA to do genome sequencing. And during the course of this work, they got a hold of this fingertip bone. It's a pinky bone that was excavated from this cave. This is Denisova Cave, located here in the Altai region of southern Russia. I mean, you can see how small this fragment is. It's not diagnostic. You can't say anything about these sorts of this sort of bone. So it sat in a museum drawer for several decades. But then when the mitochondrial DNA sequence was determined from this pinky bone, lo and behold, it was not modern human, and it was not Neanderthal. It was something else, something that had never been seen before, something that we didn't know even existed. So this is a tree of mitochondrial DNA sequences. These are all modern humans here. Here in blue are some Neanderthal mitochondrial DNA sequences. Here in red is the Denisova mitochondrial DNA sequence. So the divergence between modern human and Neanderthal mitochondrial DNA sequences is roughly 500,000 years ago. Denisovan is twice as divergent as that. Okay. But remember what I said about histories of single genes like mitochondrial DNA can differ from that of the species as a whole. And when the genome sequence was determined from this fingertip bone, it showed that the Denisovan is actually a sister group to Neanderthals. And it's a distant sister group to Neanderthals, slightly more divergent than the deepest branching we have in modern humans today. But nonetheless, it is a sister group to Neanderthals. So now we can ask, did modern humans also have sex with Denisovans when they came across them? And we can do this allele sort of sharing analysis. And when we compare two Eurasians, there's no excess sharing. Compare a Eurasian to an African, there is excess sharing with the Denisovan, just as there was with the Neanderthal. And you can see it's reduced. And this is sort of what you would expect for a sister group of Neanderthals. Where things get interesting is when you compare someone from Melanesia, someone from New Guinea or Australia. Because with Neanderthals, compare a Melanesian to a Eurasian, there's no excess matching. But with the Denisova, there is. There's excess matching to the Melanesian, the person from New Guinea or Australia, compared to someone from elsewhere in Eurasia. And that also holds if you compare Melanesians to Africans. So the simplest explanation, even though it sort of boggles the mind, is that there was Denisovan admixture, not necessarily in the vicinity of the cave there in the southern Altai region of Russia, because we don't see excess Denisovan ancestry around there, but rather 7,000 to 8,000 kilometers away in Australia and New Guinea. So the conclusion then is that humans have been having sex with multiple extinct hominins. So at least one instance of interbreeding between Neanderthals and the ancestors of all non-Africans, another instance of interbreeding between Denisovans and the ancestors of modern-day Melanesians. And that seemed nice and simple until we started doing more work and then discovered inevitably that the situation is even more complex. So with this study, which produced the first high-quality genome sequence from a Neanderthal, and it was actually a Neanderthal fossil found in Denisova Cave. So you have both Denisovans and Neanderthals in this cave. And what that showed is that when we did modeling of the history here, so if we take some modern humans, we take Denisova, we take high coverage Altai, low coverage Vindhya genome, Neanderthal genomes, and a third Neanderthal genome that's also low coverage, low quality from Mesmaiskaya cave in the Caucasus. The first thing we find when we try to model their interactions and their divergence is that we cannot explain everything we see today by the history of just these individuals. We have to invoke something else, an unknown ghost population who's extremely divergent and who contributed some ancestry to the Denisovans. Okay, so in other words, there are some genomic segments of the Denisovans which are too divergent to explain just by this single history. They imply there must be something else more divergent that contributed some ancestry to them, somewhere between 0.5% and 8%. If we should ever get lucky enough to get a genome sequence from this guy, then we could nail this down with more certainty. So we have that. We have ancestry coming from Neanderthals into the non-African populations, which we've seen before, around 2%. Ancestry from Neanderthal coming into the Denisovan at a low level. They were in the same cave. They were having sex. Denisovans contribute ancestry to Oceanians, around 3 to 6%. And then there was another signal detected of Denisovan ancestry coming into East Asians, just 0.2%, which is right at the limit of what we can detect. So a lot of people didn't believe this, but now what we are seeing with further studies is evidence of more and more interbreeding that's gone on with both Neanderthals. So as seen here are the classic two waves of ancestry here, one from Denisovans into Melanesians, one from Neanderthals into the ancestor of all non-Africans. But now we know that there was an additional episode of interbreeding that went on after the ancestors of the Melanesians separated. So Europeans and East Asians have a little bit more Neanderthal ancestry compared to Melanesians. And it turns out, curiously enough, that East Asians have the most Neanderthal ancestry at all, so that there was another episode of interbreeding with Neanderthals after their lineage diverged from that of Europeans. So multiple episodes of interbreeding with Neanderthals. We're also seeing this with Denisovans now. We see that there's at least 2 episodes of interbreeding with Denisovans in East Asians. We are also finding multiple episodes of Denisovan interbreeding in New Guinea, with the latest having occurred maybe only 30,000 years ago. And that's after New Guinea populations separated from East Asian populations. And so that implies that this Denisovan ancestry was contributed in New Guinea itself, which is then pretty remarkable that they were able to make the water crossings to get there. We're also finding other Neanderthal ancestry, okay? So Awasi 1 is one of the oldest modern human skulls. It's about 40,000 years old. It comes from this site here in Romania. It's been described as modern human, but having a few archaic features. When the genomic data was analyzed, it has a whopping 6 to 9% Neanderthal ancestry, much more than any modern human today, much more than any other fossil from around the same time. So this implies that the Neanderthal ancestry was contributed only 4 to 6 generations before this individual died. So it was practically caught in the act. And what's also interesting is that the Neanderthal ancestry in Awasi 1 is not the same as that in modern humans today. It's different. So it represents an episode of interbreeding between modern humans and Neanderthals that didn't go anywhere, that went extinct. Probably there was a lot of that went on in our past. And we're starting to find modern human ancestry in Neanderthals. So the Altai Neanderthal, this high coverage Neanderthal sequence, when it was published, it was known to have about 1% modern human ancestry. We all said, oh, that's just contamination. That's what you typically see. Until Martin Colville in the department had the bright idea of looking at this modern human ancestry in more detail and finding that it doesn't match any modern human population today. And instead, what it matches is an ancestral African population. So presumably, it reflects an out-of-Africa migration that happened before the out-of-Africa migration from which all modern populations are derived. And it went extinct, but not before contributing some ancestry to Neanderthals. Okay? And we also find that Denisovans and Neanderthals were having sex. So typically in an archaeological excavation, there will be thousands of tiny little bone splinters, which you can't say anything about. You can't even say what species they're from. There's nothing diagnostic about them. Those typically get put into a bag and shoved into a drawer somewhere. One could, in theory, try to extract DNA from those, but if you have thousands of these fragments, it's just simply not cost efficient and too time consuming to try to extract DNA from all of them. But what you can do is extract the proteins, the proteome. You can do that and analyze that very quickly and efficiently, and then assign the taxonomic category that bone comes from. In particular, you can determine whether it was hominin or not. Was it a human or archaic human versus something else? And so this technique was applied to 2,000 bone fragments from Denisova cave, and out of those 2,000 fragments, one was human, okay? As hominin, as being something in the Homo lineage, okay? And here's what it looks like. And remarkably, when the genome sequence was determined from this individual is a hybrid. It has a Neanderthal mother and a Denisovan father. So we are seeing this right in the action. And when you consider how few genome sequences have been obtained, we're seeing an awful lot of intermixing that's been going on here. And there's even some indication that the Denisovan father had some Neanderthal ancestry as well. So I think if you want to sum up what human evolution has been about, basically, it's everyone's been having sex with everyone else. That's what humans are about. Okay, now I want to finish by saying a little bit about not just that what we can learn about the sex habits of our ancestors, but that we can also leverage this information as a new source of insights into selection and adaptation that occurred during human evolution. And the way we do that is as follows. We look at the distribution of, in this case, Neanderthal ancestry across the genome. So this shows you the 22 chromosomes. The red and blue lines show you where you have Neanderthal ancestry in modern Asians or Europeans. And what you see when you look at this is that the Neanderthal ancestry is not distributed at random, that there is non-randomness to this distribution. And in particular, you find some regions of the genome which are completely devoid of Neanderthal ancestry. And these are called deserts. They have significantly less Neanderthal ancestry than you would expect. And presumably that's because the Neanderthal form of important genes in these deserts does not work well in humans, and it's been selected against. It's been removed from modern human genomes. So what kind of genes do we see in these deserts? Well, I'll just give you an example of one. One gene that's found in a desert is called RUNX2, and mutations that knock out the function of RUNX2 lead to a disease syndrome called clinocranial dysplasia, or CCD. And the features of CCD are that you get delayed closure of the cranial sutures, you get frontal bossing, I'll tell you what that is in a minute. You get abnormal collarbones, so the clavicle is the bone here, it's abnormal or even missing in these individuals. You get a bell-shaped rib cage, I'll tell you about that, and you get multiple dental abnormalities. And what's interesting about all of these is that they recapitulate some of the skeletal differences between modern humans and Neanderthals. So frontal bossing is a protuberance here of the front part of the forehead. It's not present in modern humans. It's present in Neanderthals, and it's also present in individuals that have CCD. Here are the rib cages. Neanderthals have a bell-shaped rib cage. So if you go from top to bottom, they flare out at the bottom than at the top. Humans have a barrel-shaped rib cage. It's more or less vertical going from top to bottom. Individuals with CCD, they have bell-shaped rib cages. And they also have abnormalities, as I said, in the collarbones and in the shoulder bones, which mimic what you see in Neanderthals, differences between Neanderthals and modern humans. So by looking at these deserts, we start to learn something more about the genes that were important in the differences between Neanderthals and modern humans. But in addition to deserts, we also find islands. These are regions of the genome with significantly more Neanderthal ancestry than you would expect if the ancestry was distributed at random. And so now we can ask what kind of genes are found in these islands where we have lots more Neanderthal ancestry. And so when we look at these genes, what we see is they fall into certain functional categories. Many of them are involved in the immune response. Others are involved in features of the skin, and others are involved in the diet. And what's interesting about these features is they all relate to how we interact with our environment, right? How we interact with parasites, disease organisms, how we interact with sunlight, cold, warm temperatures, and so forth, and how we interact with differences in the diet. And so these are examples of what has been called archaic or adaptive introgression. So the idea is that when the ancestors of Neanderthals left Africa, they encounter all these new environments, new climates, new diseases, new parasites, new sources of nutrition, and they adapt to these conditions by natural selection. And now, several 100,000 years later, our ancestors come out of Africa and they have to do the same thing all over again, adapting to these new conditions. But if by interbreeding with Neanderthals and Denisovans, they pick up already adapted variants to these new conditions, it gives our ancestors a leg up. They don't have to evolve this anew, new selection or that. They just pick up these already adapted variants. So I think it makes sense that we would see these sorts of variants, these sorts of traits involved in our adaptive integration in regions of the genome where we see very high levels of Neanderthal ancestry. But some of these are a little bit harder to understand. So, for example, one of the traits that varies in humans is high altitude adaptation. So populations that live at high altitudes, like in Tibet, in the Andes Mountains in South America, in the Ethiopian highlands, they have adapted to the low oxygen levels and the cold conditions and so forth by selection on various genes. And one of the genes that's been known to be selected for in Tibetans is this gene called EPAS1. And what this shows you is sort of cartoon in these different colors are the different variants, the different haplotypes of these genes in different populations from around the world. And what you can see is that in the Tibet population here, there's a very high frequency of this green haplotype. This is the one that is adapted for living in high altitude that you don't see anywhere else in the world. But where you do find it is in Denisovans. So it looks like this was something that was contributed by Denisovans. Okay, and this was rather, people treated this with some skepticism. How is it that from Denisovans you would get something that would be adapted to the high altitude? Until this find that came out last year. It's not actually a new find. The find's been known for decades, but it was analyzed and published on last year. It's a jawbone that comes from this Tibetan Buddhist prayer cave, Bashia Kars cave, up in the highlands of Tibet. It's been dated to about 160,000 years old. They attempted to get ancient DNA out of it, no surviving authentic DNA, but they were able to get ancient proteins, and these ancient proteins indicated that this was actually a Denisovan. So it's the first fossil we have attributed to Denisovans from outside Denisova cave, and it shows that they were, yes, indeed, living at high altitude, 160,000 years ago, well before any modern humans ever got there. So then we start to ask, so what were Denisovans anyway? Were they perhaps the impetus for this idea about abominable snowmen in that part of the world? Okay, so let me just give you the take-home messages that in addition to telling us about the sexual habits of our ancestors, we can use these archaic genomes to get new insights into the dispersals of modern humans, into the genetic differences that made us human, as well into alleles, genetic variants of functional importance that we receive from archaic humans. And I want to leave you with one last final thought, and that's that everything I've told you about so far is about us, right? Because we're the ultimate narcissists. We always want to know about us. We don't care about them. We want to know about us. So what did this archaic integration do for us? What did we learn about us? But, let's stop a minute and think about what the Neanderthals and other archaic humans must have been thinking when our ancestors suddenly appear on the scene, something very different than anything they'd ever encountered before. What was their reaction? Now, of course, we don't know. But there is a saying, an old saying that history books are written by the winners. And while we were certainly winners in those encounters, if Neanderthals had been able to write books, maybe they would have been writing books something like this. Who are all these foreigners with small noses and big foreheads, and where are they coming from? Look at immigration troubles in the Neander Valley. Immigration problems are an important concern all across the world today. And maybe in thinking about them, it wouldn't hurt to realize that perhaps it was the migration of our ancestors out of Africa that was the source of the first immigration crisis. So with that, I'll end with the acknowledgements. The work I've told you about has been done by a great many people at different institutions around the world, but these are the main players here in Leipzig, including our fearless leader, Svante Pääbo. And of course, we have to acknowledge the Max Planck Society for the generous funding that makes all this work possible. And thank you for your attention.
Thank you so much, Mark. That was just an awesome talk. Excellent. We've learned about our ancestry and about our past. Thank you so much. We've also learned a little bit about the continuity that enables us to ask questions about health and disease in the light of or in the context of human evolution. There is one question for you, Mark, in the chat box that says that you're talking about SNPs, and have you looked at CNVs and indels and those kinds of structural changes in the genomes? and how do they relate to the Neanderthals?
There have been one or two attempts to try to look at indels and CNVs, but the problem is that you get from the next generation sequencing, you can only, and the ancient DNA itself, you can only retrieve very short reads. And so it's very hard to get good alignments where you can really sensibly say something about CNVs and indels and so forth from this sort of data. There have been one or two attempts to look at this and try to identify, Neanderthal specific CNVs. There's at least one CNV in Europeans that seems to have come from Neanderthals, but it's technically very challenging and very demanding to do this sort of work.
There's one other question that has just come up, which is, do you have an estimate of the size of the Neanderthal population at different points of time?
Yeah. So from the genetics, we can get an estimate not of the actual census population size, but of what's called the effective population size, or the number of individuals that are contributing from generation to generation. And you can estimate that from the amount of diversity. And what we see is that actually the amount of diversity in the archaic humans that we have sequences from Neanderthals and Denisovans, it's very, very low. It's lower than the lowest human population that we have today. So it implies that they were living in very small groups, probably very structured groups. We're starting to see some evidence of that too, as we get more Neanderthal sequences. So it looks like, that there were, from the effective population size point of view, there were not very many of them.
But those were the only two questions that I can see on the chat box. And once again, Mark, thank you for accepting our invitation and for this awesome talk. Our next speaker is Mark Daly. And is Mark online? Probably so.
Yeah.
Hi, Mark.
Hi, Partha. How are you?
I'm good.
Good.
Before I move on to Mark Daly, I would like to make one more point, which is that Mark has alluded to certain regions of the human genome that can be traced back, certain regions of the human genome that are important for various kinds of diseases that can be traced back to the Neanderthal or the Denisovan. Very recently, on 30th of September, the 30th September issue of Nature, Mark talked about Svante Pääbo. So Svante Pääbo published another paper where he looked at the COVID-19 and severe COVID-19, and there was a region that was implicated in severe COVID-19, which is on chromosome 3 of the human. That has been traced back to the Neanderthal, and Mark referred to the Windia caves, and the Neanderthals of the Windia caves also had that, and it was the extent of sharing was huge, and therefore that severe COVID region, the region that enables one to get severe COVID-19 disease, can be traced back to the human to the Neanderthal. So it is very instructive to study Neanderthal genomes and Denisovan genomes and archaic genomes in order to understand our own health. Mark Daly is with the Finland Institute of Molecular Medicine, where he moved not so long ago. Prior to that, he was in the Mass General Hospital, Harvard Medical School, Broad Institute in Boston, and he's contributed enormously to the understanding of the structure, the arrangement of the human genome, subsequent to the haplotype project, and the haplotype structure of the human genome, etc. And he's been able to relate that structure to various parameters of human health and disease. In particular, he's very famous for his contribution to understanding of the inflammatory bowel disease, also for autism and schizophrenia. So those are three that stand out, but he's made all-round contributions to understanding of genomic underpinnings of human health and disease. So he's also leading or one of the leaders of the International Common Disease Alliance, to which India has become a member very recently, just days ago. And he's going to talk to us on progress of in human genetics from genomics, from genome to GWAS to medicine. And I hope that as he moves to medicine, he will talk also a little bit about the International Disease Alliance. Mark.
Thank you very much, Partha, for the generous introduction and for the invitation to come and present to this wonderful group today. It's a shame that it can't be in person, but anyway, greetings from Helsinki, where it is now at this time of year always dark, pretty much. So, looking forward to the sun returning someday. So I'm what we describe as a human geneticist, or I guess more properly maybe a human medical geneticist. And I'm going to it's a real pleasure to follow Mark Stoneking in a Symposium because I can connect now to a lot of what he has presented you already with respect to human evolution, because evolution is the main shaper and driver of the inherited component of human disease, along with some, modern natural selection, which we touch on. And so it's a great pleasure to follow him and work that I'm sure all of us have followed over many decades. My goal in using human genetics is as a tool, and it's in order to provide durable insights into the biology of disease. And ultimately, this can help us to develop therapeutics more effectively. It can help us develop diagnostic tests more effectively. And that's because the DNA that you're born with, to the extent that it contributes to disease, holds clear biological clues that are much more direct and informative, and in many cases actionable, than those that you can trace with observational epidemiology, for example. And in the lower right, what you can see is a graph of progress over more than half a century in the development of therapies for, in green, cardiovascular disease, and in blue and yellow, neuropsychiatric disease. And the difference in the dramatic ascent of medications available today for heart disease, medications available today for cancer, is that genetics has taught us fundamental mechanisms in those diseases. It has not, up until the very, very recent couple of years, taught us anything about the underpinnings of psychiatric disease, and I'll explain a little bit why. So genetics is remarkably a very old field scientifically, and there's a good 100 years of very interesting genetics research that is conducted in advance of understanding the nature of the sort of physical chemical nature of inheritance, the DNA molecule, and so forth. Yet half a century before the discovery of the DNA molecule, an actual genetic map was created of the X chromosome in flies by Sturtevant and Morgan. And of course, another half century before that, Mendel's laws of inheritance very accurately described the nature of inheritance, and that there must be some physical character that he imagined as grains of sand along the lines of pollen being passed forward. But obviously, while we had the very sophisticated understanding of the statistics of genetics and inheritance, we had no mechanism by which that could lead to any biological insights, because the field of biology was still very much in its infancy. And so we recognized for a long time in our studies of genetics over the past decades that many common diseases are very, very highly heritable. In the same degree as, or maybe not quite the same, but almost the same degree as many rare, what are considered to be genetic diseases, such as Huntington's, muscular dystrophy, cystic fibrosis, and so forth. But in fact, even mysterious diseases such as autism, schizophrenia, are extremely highly heritable, which means the DNA that you're born with is the primary contributor to why individuals ultimately would develop these diseases or not during their lifetime. Yet our ability to make discoveries about them through the decade of the 80s, through the decade of the 90s, was almost non-existent. Now, the wonderful thing about the 1990s is that it sort of ushered in an era of discovery with rather, a first generation set of tools for studying the human genome. It was only in the late 1980s that the first genetic maps of the human genome were beginning to be constructed. in a way that would allow us to trace inheritance from parents to children to grandchildren. And those tools allowed us to begin to pursue the genetics of simple Mendelian disease. And that was very, very successful for quite some time. But what was less successful was when those same tools, tracking the inheritance of disease and the inheritance of DNA markers from generation to generation, were applied to more common and complex traits, such as psychiatric disease, such as cardiovascular and cardiometabolic disease. They were not successful at all. But this, however, should not have come to anyone as a surprise. And the reason is that there had been a good century of research that had taught us about the nature of inheritance, and that it was much more complicated than simply the inheritance of a single mutation in Huntington gene or two copies of a defective CFTR gene causing cystic fibrosis. In fact, Galton and many others in the dawn of the 20th century recognized and developed a lot of the statistical tools that we use today, such as regression techniques, studying the nature of inheritance and being, for a while, quite confounded by the fact that inheritance in traits such as adult stature was very, very high. 80 to 90% of the adult stature of an individual is determined simply by the height of the two parents and the sex of the individual. Yet at the same time, there was perhaps 2 decades of debates which could not realize a harmonization of how you could have a continuous bell curve that was fully inherited, and yet at the same time adhere to the laws of Mendel, which suggested, and in many experimental systems were very validated as transmitting single factors, such as whether the pea is red or green or yellow or shriveled or smooth. And there was not a simple resolution to this. Well, ultimately there was, but it happened first through the development of experimental data, and really, a remarkably insightful experiment published in 1915, studying the length of the corolla of a flower, demonstrated that if you look at the two parental flowers, they have a drastically different length of corolla. But when you cross those and create offsprings in the first generation, cross those to make a second generation, cross those to make a third generation, what you discover is that the inescapable conclusion is that there's not a single factor that makes these two original flowers different. There are very, very many factors, and that every time you create a new combination of those, you end up with a broader distribution of offspring. And that tells us that there are many distinct inherited factors. And so this was actually the first demonstration of what we would describe as polygenic inheritance, and it actually came experimentally proven. This happened soon thereafter in flies and many, many other model systems. But it was really the statistician Fisher who put together a model that rationalized both continuous trait inheritance, as we saw in the case of the flower and as we see in the case of human adult stature, with the laws of Mendel. And it simply requires that you don't postulate a single genetic variant or factor that contributes a difference, but you propose that there could be hundreds of small genetic factors. And so with hundreds of coin tosses, considering a genetic variant to be maybe just like tossing a coin, you get an A or a B. If you have hundreds of those and each of them changes your height by a millimeter up or down, then you get exactly what you see in the natural population. And so our failure to map genes with a simple Mendelian approach, as we discussed at the beginning, which was so successful in the 1990s with respect to finding the genes underlying muscular dystrophies and Huntington's, cystic fibrosis, and just literally hundreds of other rare inherited diseases. The failure of those approaches to attack the more complex traits, such as height, for example, is very, very easy to explain, because the strategies that we were taking of tracing a single factor through generations would simply not be applicable to more complex human traits. And so the new approach that was needed and that was begun to be developed was called, a genome-wide association approach. So we could take a simpler approach to studying all DNA variants in the genome if we had the technology to do so. And of course, this was simply a hypothetical activity, a few decades ago. That sure, if we could sequence the entire genome of everyone, then we could look and find all the relevant biomedical differences. Of course, we couldn't do that, and as you heard, our knowledge empirically of the human genome was very, very crude up until the, really the late 1990s. When we first began to embark on the Human Genome Sequencing Project in the 1990s, greatly informed in our thinking by, for example, the mitochondrial work that you just heard about Mark Stoneking describe in 1987, and similar Y chromosome work that came along later. And these taught us, the empirical truths regarding the out of Africa hypothesis. One could, when we got to sequencing the human genome, and recall that the sequencing of the human genome actually took place as sequencing bits and fragments of a large number of different human genomes. And so we began to collect all sorts of DNA variants. But what you could see was the inescapable signature of the out of Africa hypothesis when we began to put these data together in the first part of, around 2 decades ago from the Human Genome Project. And what's demonstrated here is when we would find a DNA variant, that is, when we lined up 2 fragments of the genome and found a single difference between them, assuming that wasn't a technical artifact, we would begin to get the picture where DNA variation, when we would find it, was either in the center here, common to all populations in the world, or most specifically only found as a DNA variant in the African continent. And comparatively, the signature of the out of Africa hypothesis that's most, obvious is that there's very little DNA variation that's truly specific to the European continent or specific to the Asian continent. And that's, that's simply the fact of evolution. So this is quite, this gave us at an early stage a quite consistent picture. But what it allowed us to do was to sort of catalog how much DNA variation there actually was in the human population. As it turns out, humans are far less diverse than just about every other species that walks the planet, with the exception of some that are extremely endangered now. But moving forward from that, this was a well-established fact in population genetics, many, many decades ago, and we're simply providing empirical evidence of that at this point in time. And so if you take the idea that most genetic differences have this sort of pattern of being common across all populations of the world, most genetic variation that contributes to common human traits and diseases should also be explained by this. What was a particularly important step towards progress in this field was that we didn't have to sequence the entire genome, and we didn't even have to ultimately look at anywhere close to the 10 million DNA variants that were common across the world. And that was because another feature began to recognize itself. Because human evolution is, in evolutionary terms, very, very recent thing, and the dispersal throughout the world, very recent. Mutations that we find, when we find a DNA variant, more often than not, that DNA variant that we've discovered in an experiment in the lab today is one that has a unique origin somewhere in human evolutionary history. And so the consequence of that is that when we find multiple individuals that carry on a single chromosome the same unusual DNA variant, we can identify segment of DNA for sometimes millions of bases, sometimes hundreds of thousands of nucleotides to the left and right of that location that are identical to each other, that don't show variation from between those two individuals. And that's because what we've identified is we've identified a mutation that has occurred at one point in human history, then been passed down through separate tracks to those two modern individuals. And we can recognize the identical sequence of the founding individual and the founding chromosome on which that mutation occurred. This was well established to be an effective technique for discovering rare mutations for relatively rare diseases, even if those mutations had happened thousands of years ago. For example, in the cloning of the cystic fibrosis gene, the little map in the background demonstrates, uses color to represent identity of two chromosomes from a cystic fibrosis patient around the CFTR gene. But what we were able to demonstrate, starting, in this at the end of the Human Genome Sequencing Project, was that this was a general pattern, and that while there were millions and millions of DNA variants, there were actually far fewer, sort of distinct sequences or distinct haplotypes that happen to be quite diverged from each other, as a consequence of the fact that recombination in the human creates new assortments during meiosis in the creation of sperm and egg of each individual chromosome. This was happening only at infrequent positions along each chromosome. And so these patterns gave us a path forward that we followed that led us to genome-wide association studies. First, however, though, recognizing the fundamental nature of human genetic variation and these patterns of through the recombination and mutation coming through evolution, in order for us to apply these in the development of techniques for studying human disease, there had to be many technical and accomplishments along the way that enabled us to apply technology at scale. And these first technological advances came with genotyping technology that would allow us to look simultaneously at thousands and then tens and hundreds of thousands of specific known DNA variants in a single experiment. And then ultimately with the advent of DNA sequencing technologies that were massively parallel in nature. And so the idea that we could study a polygenic trait, so study a disease that causes, is highly heritable, but has a very distributed genetic origin, was not a new idea by any stretch of the imagination. In fact, there's a very perceptive paper 50 years ago describing this concept. And for decades, this general concept that heritability of traits is explained by genetic factors scattered throughout the genome has underpinned animal breeding activities, for many, many years. So the genome-wide association study came online and emerged in a very, sort of clear way starting around 2005. And since that moment, when we could begin to study even hundreds of cases of a disease and compare them to hundreds of individuals without disease, sort of the simplest of all epidemiologic designs, with a genetic component, this has been the primary driver of discovery of genetic variations underlying common human disease. I'll give you some examples from inflammatory bowel disease, Crohn's and colitis, which is a disease that has been on a rapid ascent throughout the world in the last 50 to 75 years and continues in almost every, with, a development or in all parts of the world, the rate of IBD, as with things like type 1 diabetes and food allergies and so forth, is on a significant ascent. So IBD has had great success in terms of the discovery of genetic variations underlying disease, and I'll come to why this is important and how this is being used in a short time. the first efforts to apply this to psychiatric diseases and brain diseases of all kinds were not successful at all. In fact, we did some substantial studies in the latter parts of the last decade, which were not at all informative and didn't discover any genetic risk factors. And so we scratched our heads and thought a bit about why that might be when they were equally, if not more, heritable than many of the other diseases that we were studying. And of course, the role of natural selection. So while we think to this point of the sort of neutrality of DNA evolution, recombination, interbreeding, as you heard from Mark, as shaping the genetic architecture of humans, there's of course natural selection directly acting against phenotypes. And in fact, some of the traits that we were studying, schizophrenia and autism in particular, have some of the strongest evidence of natural selection outside of truly lethal genetic diseases that exist. And in fact, individuals with these diagnoses have far fewer than half the number of children as individuals in the rest of the population. This doesn't mean that the approach we were taking couldn't succeed, but what it did mean was that it would require a much greater scale in order to succeed, because natural selection is simply putting a cap on how large an effect can get to a certain frequency in the population in terms of a genetic variant arising as a mutation and then causing a certain risk to disease. And so global consortia began in psychiatry, in cardiometabolic disease and inflammatory diseases across the board as a path forward to, rather than competing with each other to discover a gene, we would work together and have a chance to discover, in fact, hundreds of genes, as it turned out. And so the evolution of schizophrenia studies is shown here. And by the time we had reached 2014, we had discovered more than 100 distinct regions of the genome with genetic variations associated to risk or protection from schizophrenia. So now we suddenly went from not being able to discover genes to, if anything, being able to discover genetic variations too easily, because this creates the next challenge, which we really have to focus on, which is how do we do something with all of this information? In no cases have we found any violation of that force of natural selection. It simply required a much larger sample size than we initially started out with, because the effects of individual genetic variants are extremely weak, because if they were strong, they wouldn't be permitted to exist by natural selection. And so in the case of schizophrenia especially, we required tens of thousands of cases to really make almost all of these discoveries. But the real point of the whole thing is to try to get insights into the biology of disease from genetics. So it's not simply to create a stamp collection of genetic variants and say, oh, I found more than you did. It's really to gain new insights into disease biology that we might be able to convert into some real new ideas for therapeutic development. So I go back to IBD and describe here some of the work of my colleague Ramnik Xavier. But the burning question that we've been wrestling with the last few years is that, okay, we can find hundreds of genetic variants in these diseases, but it's very, very difficult to move from a genetic variant to an actual function and a mechanism that would give us an insight into what's actually causing the disease. The DNA marker itself is not a useful, we can't treat the DNA variant in any real way. And so without reliable mechanisms to connect these genetic discoveries to individual genes and molecular mechanisms of risk, there's no way we can actually make good progress in understanding and treating disease. And so we have addressed two possibilities. And this comes to the International Common Disease Alliance that I'll mention in closing. That there are instances, the majority of instances, the DNA variant that we've discovered here, this little purple dot, is not specifically affecting the protein coding of a gene. And so it doesn't immediately even tell us what gene might be implicated and how. And so there's a huge amount of unknowns with respect to the regulation of genes, the creation of the translation and transcription, creation of proteins from DNA. And as a result, we are not yet very good at reading the information from these, and we need a lot more work to come to make progress on this. The technologies for doing, for making these inferences are really still in their infancy. This was a paper just published last year. And as you can see from the numbers here, we're only at a point where we're really able to apply to very small numbers of individuals the most detailed dissection of the cell types and their expression profiles and so forth in specific tissues of relevance to specific diseases. So what we have found is that despite having great success in discovering genetic variants, the vast majority of the genetic variants we find are difficult to interpret, and so that greatly limits their utility. On the opportunistic side, though, there are some number of these that point directly to protein coding changes, and here the genetic variant is telling you what it's doing to a specific gene and therefore a specific protein. And this gives us a much bigger leg up, as I'll show you, on experimental activities that follow downstream. Those genetic variants that we discover with the GWAS technique that affect the protein coding of a gene also have much bigger effect sizes on risk, and so they confer more risk. And so biologically, they're going to, for all of these reasons, be more important and more informative about the biology of disease. And so many examples, and almost all of the examples that you hear about with respect to genetic discoveries leading to novel therapeutics fall into this category where the genetics pinpointed specific variants in specific genes that allowed a set of biological experiments to follow in a very targeted way. And so we have seen this in our work in IBD, and in fact, with several of these variants, we're able to, at an early point in the process, target new pathways that had not been previously suspected of having a role in inflammatory bowel disease. Yet, just recognizing those genes for a geneticist with a statistical background, such as myself, was not helpful really to the biologists or the clinicians treating IBD. And so you need to partner with experts such as my colleague Romnick, and then we begin to move together across this bridge of what it is we need to accomplish in order to gain insights into the biology of disease that are actionable. And so he and his colleagues have certainly done so over the last decade with respect to that original observation from our IBD study, identifying specific cell types, modeling them in mice, and then moving all the way to testing new chemical compounds for reversal of the very specific mechanism that they discovered. And that's really a pattern that we try to now repeat over many of our discoveries in IBD. I'll skip through a few of these examples, but with the advent of new DNA sequencing technologies, by 2013, we were able to now begin to sequence a genome for a few $1,000s. When the conduct of the Human Genome Project, we were still putting together 10 years earlier, one human genome for roughly $1 billion US. And so this brought us to the point where we could now really ramp up the discovery activity for even rarer and higher impact mutations. So remember, those high impact mutations are kept to low frequency by natural selection, but now we were able to unlock them with the sequencing technology and discover, for example, even in some cases, a sort of a specific mutation that truncates the end of this gene, CARD9, with the sequencing technique, and causes, in this case, a strong protection against the development of Crohn's disease and ulcerative colitis. And so that gives then a very clear clue and pointer by which, if we know a specific mutation is protective against the development of disease, Romnick and his colleagues can then go with a very targeted tool and identify exactly what the function of that mutation is, and then begin to develop chemical compounds that mimic that exact function. This gives you an assurance that the drug that you are aiming to develop would have a natural efficacy and would be safe, because the natural genetic variant in individuals walking around is proof that it can be both safe and effective. And so we embarked in a deep approach starting about 5 years ago to use sequencing technology to really advance our understanding of Crohn's disease and ulcerative colitis, sponsored by charitable trusts and government funding agencies. And this has led to a number of additional new findings with respect to specific genetic variants that give us clues, building blocks, if you will, to the biology of disease, but also give us potential, in a number of these cases, targets for therapeutic activity that would be proven to be effective in preventing or ameliorating IBD. And so this is very difficult work, and the consequence of and the result of highlighting that is that it takes a huge amount of effort by extremely expert groups to pursue even one of those genetic discoveries to the point of delivering concrete biological insights that might have a remote chance of delivering something effective therapeutically. And so this challenge is really what underlies our efforts to found with a global community the International Common Disease Alliance that, as Partha introduced, India has just joined, and we're very, very excited about that. And the underlying challenge is, as I've laid it out in this talk, we've essentially solved gene discovery. So if you have a genetic or inheritable disease, we know how to find the genetic variants that cause that disease. But what we don't know is what to do beyond that. And what to do beyond that sits in this realm of, after we discover genetic variants, they must be affecting the function of a gene, but we don't know which one. It must be a relevant function to disease in certain cell types or affecting certain cellular functions, which are also unknown. And then ultimately, that change, that molecular change, is somehow conferring risk or protection from the development of disease, and as a result, it has a relationship to a physiological function, which is also unknown. So there's a lot of unknowns, unfortunately, to go from a variant to a disease. And so that's the global challenge that we've set for ourselves with the ICDA, working together as a global community to sort of unblock the path from discovery of a genetic variant that affects disease to understanding the biology and delivering on the medical value of that. And so this group has been meeting periodically over the last couple of years. Here's a picture from sort of an official launch meeting that happened in the fall of 2019. And we have, at that meeting, sort of initiated an organizing committee with global representation, and thanks to Partha for agreeing to join and help move this activity forward, to plan out the next steps that would be required in order to create both an effective global research community, but one that really emphasizes the importance of acting together globally to advance not only discovery, but also equity and assurance that the results of this research are valuable to all people of the world. And so there's a white paper with recommendations. You can read it if you are interested. And the main focus areas will be many, but they sort of start walking all the way from the genetic discovery, which is well in hand for many diseases, through to the delivery of medicines, but also with a significant and important emphasis on assuring alongside this set of scientific activities, there is a proper attention on global equity, policy and ethics, and innovation and training worldwide. So from my research standpoint, being on the genetic side, we are advancing through this mechanism, hopefully, many activities such as propelling the genetic analysis across biobanks globally rather than individually, cataloging genetic variation of all populations of the world and making that a public resource, and then developing standards such that the results of genetic studies can be broadly used by researchers around the world. And as a final note, one of the offsprings of the type of organizational work that we've been putting in here in the ICDA and also just over the past years in these collaborative efforts have been that we were in a good position to help initiate back in March at the beginning of the global phase of the pandemic, what we call a COVID-19 host genetics initiative. And this very rapidly grew to a truly global activity in which research groups around the world, if they were performing human genetic studies of individuals with a severe response to COVID-19 SARS-CoV-2 infection, could work together in a network to advance gene discovery, which might give us some clues, ideally, to places where we might repurpose drugs efficiently. And so we've had a great response to this with participation from many established biobanks around the world on the left side, and on the right side, many new studies that were initiated in response to COVID-19, as well as several direct-to-consumer companies who are contributing freely to this activity. And the only rule of the road in this activity has been that any analysis that the group does together, the results are made public immediately on the web so that anyone who might benefit from them, whether they are an academic researcher, a pharmaceutical company, can use the latest and greatest genetic insights that might emerge from this. And some insights have emerged satisfyingly in the last version of data that we posted. There were now a number of truly robust genetic findings that have genetic variants, and one of which, the tallest one here on chromosome 3, as Partha mentioned, originates as a variant from the Neanderthal population. But these are genetic variants which, by and large, help, explain some fraction of why individuals who are infected with SARS-CoV-2, why some of them have a very severe and life-threatening respiratory infection, while others have little or no symptoms at all. And that's been the main focus, and the hope is that some of these provide clues to where there might be effective repurposing of drugs. And so I thank Andrea for all his efforts in launching this activity, and I've shown you all those. And just want to summarize, then happy to take any questions, but I think this captures the main points that as you heard from Mark Stoneking, human diseases, as well as just the neutral patterns of variation, have a very significant contribution from genetics through both the natural evolutionary history of humans and, as well in some cases, the strong forces of natural selection acting against the disease in the modern times. And over the past decades, from the sequencing of the Human Genome Project to the current date, we've really made a routine business of discovering what the inherited genetic components are for both common and rare disease. However, moving from those discoveries to medicine is a much more challenging task, and this is going to require a new level of technology and global collaboration, much as the progress that I described in DNA studies were able to be revolutionized 10 to 30 years ago with technologies and insights into the human origins of DNA. We now need to do that in the much more complicated space of biological function. So thanks for the invitation again, and happy to take any questions.
Thank you, Mark. Thank you very much for a wonderful talk. I must first of all, I'm reading on the chat box, I must first of all state this, that one of our most eminent clinician researchers, a very eminent endocrinologist, a diabetologist, V. Mohan, has complimented you for a wonderful talk, for a great talk. There are two questions.
Thank you very much.
Yeah, thank you. There are two questions that I see, Mark, on for you. One is that following up on GWAS, have there been many studies on many diseases using CNVs and other kinds of structural variants in order to understand what role those play in diseases? That's question number one. And question number two is, in terms of monogenic diseases, can common variants explain the differences that we see in pathophysiology in a particular disease?
Great question. So yes, so sorry to gloss over it. I mean, our genetic studies actually started in many cases with CNVs because they were far easier to recognize and to type back in the early days, at the beginning of this genome-wide association activity. And in fact, they continue to play a component of the role. Many of our first discoveries, especially in autism and schizophrenia, as well as in many developmental diseases, were large deletions or duplications of DNA, and they continue to play an important role. However, the challenge there is not unlike the one that it's different than the one we have with the single nucleotide variants. But the challenge was that we discover CNVs that confer a strong risk to certain developmental disorders, but they've been challenging to extract biological clues from conclusively, often because those deletions or duplications are deleting or duplicating 25 or 30 genes. And so it's been very challenging to move to biology without understanding which of the genes is specifically relevant. But for sure, in all of our studies, the CNV component is right there alongside the single nucleotide variant component. And then the second question was a very good one, and one that we very actively are addressing in many cases, and that is that do you find the variability in presentation in the case of a sort of Mendelian disease to have genetic underpinnings? And I think there are a number of cases now that begin to emerge where that is the case. And in fact, we see in both in cancers that are driven by strong familial mutations, as well as in psychiatric diseases that often have a strong, acting mutation precipitating the disease, we see that the polygenic background actually has a significant effect on both whether the individual manifests the disease and sometimes at what age they manifest the disease. And so I think, this is starting to become a more common observation. The challenge is that, in most of these cases, that it's quite difficult to collect a sufficient number of individuals that carry a specific mutation or a mutation in a specific gene that confers a high risk to disease. So to actually conduct those studies conclusively is not so easy, but I think it is definitely a high priority topic that we focus on now.
Thank you again, Mark. There are no other questions. There's a question from YouTube, and this is a question that says, how about, this is for Professor Stone King, not for you, Mark. So we might come back to Mark's questions if we have time left at the end. Mark, we look forward, nationally, Indian scientists look forward to engaging with you and your team in the coming days now that the Minister of Science and Technology has kindly approved participation in the International Common Disease Alliance. We not only look forward to a collaboration, we look forward to seeing you in India as soon as the pandemic sort of goes away or becomes an endemic to the world. So yes, thank you very much, and we look forward to stronger ties with you and your team in the coming years and days. Thank you, Mark.
I look forward as well. Thank you, Partha. Very appreciative.
Thank you so much. Our next speaker is Dr. Arindam Maitra. Dr. Arindam Maitra is my colleague at the National Institute of Biomedical Genomics. a great friend. He is the person who actually helped me enormously on two counts. One is initiating the International Cancer Genome Project in India, and the second is building or establishing the National Institute of Biomedical Genomics. I'm very grateful to him for his enormous contributions to these two activities in particular, and many others in general. Arindam graduated from the Calcutta University, did his PhD at the All India Institute of Medical Sciences, served in the industry, got bored with the industry, went and joined academic institutions. Finally, he joined the National Institute of Biomedical Genomics, I think in 2010. He was probably the first faculty appointment that we made in the National Institute of Biomedical Genomics. He is going to talk to us on genomic basis of oral cancer, something that we've been doing as a part of the International India project of the International Cancer Genome Consortium. Without further ado, Arindam, we look forward to your talk.
Many thanks, Parthada, more formally, Professor Majumdar, and I thank you and the rest of the distinguished organizers of the INSA who invited me kindly to participate and share this dais in front of a very distinguished gathering. I will just share my slides. Can you see my slides?
Yes, Arindam, they are visible. If you can make it full screen.
Yeah, perfect. So as I said, so today I'll be talking to you about the contribution that Indian participation did to the study, which is also known as the 25K or the International Cancer Genome Consortium. It had been a privilege to actually work with Portugal from 2009-10 to start this activity of this International Cancer Genome Consortium, the first large-scale genomic study of an international collaboration that we initiated in India, and where we used various methods and cutting-edge genomic technologies for the first time in India, actually, and then go on and also build the institute. So why do we study cancer? Because cancer is a very important disease. 1 in 3 develop cancer, and actually 1 in 5 people actually die of cancer. So there are many, many different types of cancer. And what our understanding of the recent years, and particularly the last decade, has shown that it is essentially a genomic disease. It's caused by various alterations in the human genome, and which results in the aberrations in the cell phenotype, which results in cancer. The challenge, the real challenge that in treating cancer has been that in addition to the various different types of cancer, in a particular single type of cancer, even now we understand that every tumor is different. It's caused by its own set of mutations, and itself is it's as if it's a single disease. And so every cancer patient is different. So understanding this over the last 10 years and then trying to find out what are the commonalities in this diversity has been the major challenge for the international community. Essentially, all the research on the cancer genome has been focused on 3 different questions. The major 3 different questions. The first question is, what are the genes that are mutated in a particular tumor? The second question would be that what are the processes which causes mutations in these genes in this particular cancer? And the third is how these mutations result in transformation of the normal cell to a tumor or a cancer cell. So as we understand now that we call cancer a genomic disease, essentially because all the genomic alterations are the basis for causing cancer. But out of these genomic alterations, only a handful of the mutations that are in the germline are those that are inherited from previous generation. Vast majority of the mutations are occurred during the lifetime of an individual, and it's a evolutionary process. It starts from the fertilized egg, and as the cells multiply, through several rounds of mitosis and they continue during one's lifetime, they start acquiring mutations due to the error in our replication machinery. And these errors generally accumulate. Most of these errors are selectively neutral. They do not confer any selection advantage to the cell which harbors it, and hence they just keep on acquiring and getting passed on from first one generation of the cells to the next. And these are termed as passenger mutations. Only a handful of the mutations, rarely a mutation causes a selection advantage to the cell that harbors it. And there are many minor examples of these minor winners of this competition, like many of us harbor skin, we harbor moles on our bodies, and these are examples of these minor winners of the competition. Very rarely would a mutation result in invasive property of the cell, transform the cell from its normal phenotype to one that goes in for uncontrolled cell division and finally results in invasion. Now, these mutations are rare. Thankfully, they are rarer and reside in a finite number of genes. And because these mutations, once acquired, drive tumorigenesis, or as we say, the formation of the tumor and then progression of the cancer, these are called as the driver mutations. So they are very heterogeneous, and there is, as I said, there's a commonality in diversity, and we'll elaborate on this as we move ahead. Now, so the reason why we had learned so much about how the genomic basis of cancer over the last decade has been this disruptive change in the way we can investigate and identify these mutations in the genome. So for ages, we have been doing sequencing methods which are based on Sanger, and we used to look into, so it comes as a peak, and we see peaks under peaks as mutations which are somatic in nature that we are trying to identify. And many times it's very difficult to identify these, and because of lack of sensitivity and quantitativeness of the sequencing data that we used to do. Until 10 years back, this platform, this methods changed, and now we used what is known as collectively as next generation sequencing, and these methods are continuing to evolve. In these methods, other than making enormous leaps in terms of throughput or speed of sequencing and cost of sequencing, which continues to go on and making things very favorable to sequence individuals, one of the biggest advantages of this method, which benefited cancer genomics, has been the quantitativeness of the data. Now we no longer look at peaks, but we look at reads. And in these reads, we can count in a particular position a mutation occurs how many times or what proportion of the reads harbor the mutations. That enables us to do statistical tests to understand whether there is a mutation that's there and identify all the somatic mutations which are present in a particular tumor cell. So with these technologies, the whole world started take on various kinds of cancers and to identify the somatic mutations that drive these cancer types. So essentially in all these kind of studies, the way of approach doing is take tumor and take a normal tissue. Generally for solid tumors, it's blood. And then we extract DNA from them and we perform whole genome or exome or sequencing. And so you understand that the mutations that we don't see in the normal tissue, but we see in the tumor are the somatic mutations, and then we focus our interest on these somatic changes. Similarly, looking at expression, one can look at, instead of DNA, one can look at RNA from the tumor and that just in normal tissue. And again, similarly, again, DNA for DNA methylation changes. So with the advent of this technology and the methods, the international community decided to launch a big offensive against cancer, and this resulted in the formation of the International Cancer Genome Consortium, which sequenced about 25,000 cancer genomes, and that's why it's called the 25K initiative. The consortium proudly claims today that no cancer therapy is developed today without the genomic knowledge that ICGC provided to the world. The purpose of the ICGC was to obtain a comprehensive description of genomic, transcriptomic, and epigenomic changes in 50 different cancer types, which makes it 500 tumors per tumor type, and then that lands up with 25,000 genomes. So it's a big consortium, had various groups which specially focused on different aspects of the work before the entire initiative started, and created SOPs and harmonized protocols through which all the participating centers followed the same to get results which are comparable. India is a founding member of this consortium from the first days of the consortium, and the Indian project has two partners. The clinical partner is the Tata Memorial Center. The team is led by Dr. Rajiv Sareen. The genomics partner is the National Institute of Biomedical Genomics, where I belong, and Professor Partha Majumdar is the leader for that. And he is also the national coordinator of the ICGC India initiative. This is one example where, for the first time, the funders participated in the consortium. So the secretaries of Department of Biotechnology, Government of India, at that time, Professor M.K. Bhan, and then with his deputy, T.S. Rao, they participated in so there was a scope for funders to participate, like an oversight committee, and actually monitor and make suggestions in the activities of the consortium. So India took on a particular cancer type, which is known as oral squamous cell carcinoma of the gingivobuccal complex, or the OSCCGB. Now, oral squamous cell carcinoma, or the oral cancer as we call it, is the 8th most common cancer worldwide. And the reason why India took on oral cancer was it is the leading cancer among men in India. Almost about 260,000 new cases and 128,000 deaths occur annually. Users of tobacco have nearly 30 times higher risk of developing this particular cancer. And in India, we have the grand concoction of tobacco with or without combination of betel leaf, areca nut, and select lime. That is the primary cause of this cancer. Another big alarming reason why this cancer needs to be combated in India is that a large section, a substantial section of the younger age boys and girls, less than 15 years, have also habits of tobacco chewing, which is the predominant risk for this cancer type in Indian subcontinent. The OSCCGB, so this is the gingivobuccal oral cancer, as we call it, is a unique feature which predominates in the subcontinent as compared to the West. If you look at the differences in the site of the oral cancers, in the West, it is predominantly tongue and floor of mouth. Whereas in the Indian subcontinent, it is predominantly the gingivobuccal complex, which consists of the buccal mucosa, the retromolar trigone, and the lower gum. So this is a unique feature. That's why OSCCGB was the cancer that we undertook to investigate and elucidate in the Indian initiative of the ICGC. In case of OSCC in India, the surgery with or without post-operative adjuvant therapy is the gold standard of treatment that we all know. So what we did in the project was we recruited patients. We took their biopsies, surgical resections of the tumor, and we also took blood from them for the DNA. And for RNA extractions, we also took adjacent normal tissue. We undertook whole genome, and initially we started with whole genome sequencing. But as the next generation sequencing technologies evolved with more firepower and more accuracy, we went on to whole genome sequencing like the rest of the consortium and other genomics initiatives. We also use SNP single nucleotide polymorphism based microarrays, whole genome microarrays, to look into, one is to QA/QC the data, and the second was to look at copy number variations. We also, in addition to the nuclear genome, we also undertook analysis of the mitochondrial genome, and we used RNA sequencing and methylation arrays, whole genome methylation arrays, to look into gene expression and DNA methylation data. So this is the mutation spectrum that we first reported for OSCCGB. The striking thing that comes out and that was at that point of surprising to us was that there is a wide variation in the number of mutations every patient had. I mean, there was a big variation. So we looked at the coding region of genomes of the 50 patients initially, and we did to multiple platforms, to orthogonal platforms to validate the verify the findings. We found about 5,600 somatic variants, of which about 3% of these variants are only indels, and the remaining all were single nucleotide substitutions. Out of these single nucleotide variations, we found about 25% of them were synonymous, and the vast majority were missense mutations, along with small proportions of nonsense splice sites, mutations at the splice sites. Among the indels or insertions and deletions, frameshift deletions were the most common, almost half of the indels that we found. The mean number of variants that we found per patient was around 113 if we included the synonymous variants, and where if we excluded, we looked the synonymous variants, it was around 85. But the interesting feature was the range. In the former, it was 13 to 939, and the second, it was 12 to 637. The next interesting feature is the mutation rate. The way, so in the ICGC, it was observed that different cancer types have different propensity to mutate somatically. So this is one snapshot from the data, ICGC data portal, where the mutation rates of all the cancer tumor types have been put in a single display. And if you look, the lowest mutation rates are those from the pediatric cancers or the leukemias, whereas the highest mutation rates are those from the melanomas or the skin cancer cases. The oral cancer that gingivobuccal oral cancer had medium kind mutation rates. The median was around 3.6 mutations per MB of the genome. This was based over about 178 participants at that time point when we calculated it, with them based on 26,000 mutations. So we also looked into, so historically earlier, human papillomavirus infections have been found has an interesting role in head and neck cancer. There were reports of HPV, particularly more from the TCGA and from the West, of involvement of the human papillomavirus infection in head and neck cancer. We looked into our data sets using various approaches, like trying to detect HPV leads in our sequencing data, and then also undertaking PCR-based approaches. We did not find any statistical significant difference in the proportion of different mutations in patients with or without HPV. We did not even find, so there was mostly there was no relationship with HPV infection, and there was no significant difference in the mutation rate between HPV positive and HPV negative individuals. So by now, the entire community now understands that in the Indian subcontinent, probably HPV is does not appear to play that kind of significant role in oral cancer. The chewing of tobacco is the overpowering and sustaining cause for this cancer type. We looked into the mutation signatures. So different mutagens or the background processes that cause particular mutations can be identified in the genomic data. And we found that most of our patients were exposed to tobacco had C2A and G2T transversions at a very high rate. About 61% of the patients had this mutation. This is compared to 15 to 26% patients who have these mutations in other cancers not associated with tobacco. And this proportion is about 31% in the general population. So there is a very strong signature of tobacco-induced C2A and G2T transversions that we found in the OSCCGB tumors in this place. This tobacco signature, we again looked deeper and found that these tobacco signatures were more frequent at a particular context of the sequence. So we found that when the C is in a context of upstream G. So it's a GCX, where X can be any of the three bases. So the GCX has a higher propensity to mutate. So the C to mutate to A. So GCX to GAX, that's the highest pattern of mutations that we found in OSCCGB. We also picked up some overrepresentation of C2G and C2G mutations at a different context, which was like 5' TCX. These kind of mutations are also seen in other cancer types like breast cancer and are associated with an overactivity of ApoBec family of genes. We also found that the C2G mutations were predominant at non-CPG sites, contrary to what is normally observed in the germline. In germline, you find these mutations more in the CPG sites. So this is a picture of the entire ICGC data set. So if we collate all data from all different tumor types, and we find what are the recurrently mutated genes. So these are the genes that come through on the top. So it's the skyscraper that you see here is the TP53 gene, which is very well known, and that's widely known to be the most commonly mutated gene in various cancer types. And then you have a whole series of other genes. All these genes belong to one or other of the hallmarks of cancer that is widely known. When we looked into this is the OSCCGB mutation pattern, you see that again the pictures is somewhat similar, which is not surprising. The TP53 is the predominant mutation, a mutated gene in this particular cancer type. But we also have other genes like the caspase-8, FAT1, Notch1, and a whole series of genes. So even in the top, the genes which are most commonly mutated in various cancer types. In towards the middle, you have genes which are more unique to a particular cancer type. And then you have a long tail, like the tail of the T-Rex. And this tail hides all the genes which are more private in nature. So that gives a clue to the fact that that's why one reason why the deep sequencing of genomes of tumors have resulted in so much information on genes which are unique to a particular cancer patient. And because of this, the cancer genomics has seen the biggest application of personalized treatment by translating this genomic information that has been generated over the last decade. So we looked into the other genes that we, entire spectrum of genes in the coding region, and we found that about 4,100 genes were mutated, out of which about 980 genes were mutated in more than 2 tumors. And 45 of the genes were mutated in better than 10% of the tumors that we sequenced. And most of these mutations were non-synonymous, deleterious, or damaging. When we looked at the mutated genes profile, we found different kinds of information. We found that the OSCCGB Harvard were driven by genes which are also common and reported earlier for other head and neck cancer types. These were the TP53, FAT1, caspase-8, HRAS, and Notch1. We also found amplifications in genes CCND1 and TP63. These are again, and homozygous deletions in GSTT1 and CDKN2A and CDH19. So these are all known drivers of HNSCC. But we also found unique drivers of OSCCGB, and these were a handful of genes that were very prominent: USP9X, MLL4, ARIC2, and then UNC13C and TRPM3. We also found unique copy number alterations in certain genes, like amplifications in DROSA, YAP1, MECOM, and NF1, PSP1, and the MMP gene cluster. We also found homozygous deletions which are unique to OSCCGB in DDX3X, and then also we had deletions in CCNC and PUL. We also found mutations in other cancer-associated genes, like SYN1 and SYN2 and so forth, but these did not reach significant levels, but there were patients who had mutations in these genes as well. When we mapped these mutations to the pathways, we found that there were significantly mutated pathways which had been common to other head and neck cancer phenotypes. These included P53 signaling pathway, apoptosis pathway, PI3K AKT signaling pathway, and then NOT signaling pathway. We also found that there were unique pathways which are mutated in OSCCGB. These included neutrophil signaling, Wnt signaling, dorsoventral axis formation, and axon guidance. So we found in our mutation data, and when we mapped these mutations to pathways, that there were genes which were mutated genes belong to either those that were predominantly mutated in other head and neck cancer types, but there were also genes which were unique to OSCCGB. So we use these mutations to understand how these patients cluster, and we found that there were interesting molecular subgroups. And these molecular subgroups, we found that when we did clustering, these mutations led the patients to be clustered into 3 predominant subgroups. The first cluster of patients belong to C1, where patients were unique feature was they were mutated in either caspase A, PAT1, Notch1. The second group, second cluster of patients were dominated by mutations in the TP53. And the third group were contributed by mutations in a heterogeneous set of genes, but predominantly with USP9X and or MLL4. Now, other than this, just the clustering, we also found that there was an interesting feature of these molecular subgroups. We looked into what is known as the disease-free survival. That means after the surgery is conducted on a cancer patient, what is the time period over which there is no further recurrence of the disease? That was measured in months. And we found that the disease-free survival in the patients varied from 1 to 39 months. So quite variable, and with an overall mean of 14 months. Now, we found when we compared the disease-free survival of patients between belonging to these different clusters, we found that patients belonging to the first cluster, which is those with a caspase 8, Notch1, FAT1 mutation, and those who belong to third cluster with MLL mutations in MLL4 and a combination of other genes, had significantly longer disease-free survival compared to those who belong to the second cluster, which is a cluster of where the patients have mutated TP53. So it shows that the TP53 mutated patients had shorter disease-free survival compared to these 2 other clusters. We also found that patients with mutations in MLL4 had a significantly elevated duration of disease-free survival compared to those who did not have mutations in MLL4. So these were unique features of these molecular subgroups which are associated with clinical features of the patients. Now we looked into certain other pathways. So when we, one particular pathway that is very important is the arachidonic acid pathway. Now, what we found was that arachidonic acid is a key inflammatory pathway, and this has been implicated in other cancer types. The pathway generates genotoxic agents and reactive oxygen species that actually promotes tumorigenesis, or formation of the tumor and progress of the tumor. So major mitotic signaling pathways, including PI3K and MAP kinase and Jun kinase pathways, are triggered by promoted by the products of this arachidonic acid pathway. There are some reports of its the involvement of the arachidonic acid pathway in HNSCC, but there was there was scarce. There was no information of the gingivobuccal oral cancer. So inhibitors, we know that inhibitors of arachidonic acid metabolism pathway has shown promising results in treatments of various cancers, and there are some clinical trials at the time that were ongoing. So when we looked into, we found in our patients, we found that the mutations, actually there were patients, multiple patients who had mutations in the various genes which belong to pathway. These genes either belong to arachidonic acid production or the COX component or LOX and CYP component of the pathway. When we compared patients who had mutations in these genes belonging to this pathway versus patients who did not have mutations, we found that if patient had a mutation in one or multiple of the genes in this pathway, had a higher disease-free survival. So they survive longer, more than 12 months after surgery, compared to if the patients did not have mutations in this pathway. So we looked into the survival statistics and we found that the mean disease-free survival of patients who harbored mutations in the arachidonic acid pathway was about 13 months, 24 months compared to those who did not harbor the mutations, where the mean disease-free survival was about 13 months. So to validate these findings, what we did was we pulled our data with the data downloaded from the Cancer Genome Atlas head and neck cancer dataset, and we found that these results held on even in the pooled larger dataset and was verified and validated. So knowing that, so we now know that arachidonic acid pathway probably plays a very important role, and this shows a lot of promise that inhibitors of arachidonic acid pathway can actually be a good example of how a repurposing of drugs can be done to handle gingivobuccal oral cancer. Another aspect, shifting gears slightly, another aspect of OSCC-GB is the lymph node metastasis. It's an aggressive phenotype and it's a strong prognostic factor, closely associated with reduction in survival. Now, this ability to invade the lymphatic system is a property of the head and neck cancer, and it's unlikely to be determined by other factors like primary tumor size. And there are some reports of gene expression alterations and deletions in lymph node involved in lymph node metastasis in various cancer types. So we decided to look into what are the genomic factors which are associated with lymph node metastasis in OSCC-GB. We found that, cutting a long story short, we did whole genome sequencing, whole exome sequencing, and array copy number based array-based copy number analysis of paired tumor and blood data. There were no significant difference in other aspects like age, gender, mutational landscape, and average mutation rates between patients. Two groups of patients: one, patients who had the highest tumor stage like T4 without lymph node metastasis, and patients who belong to other stage, but they all had lymph node metastasis. Either at least two histopathologically confirmed metastatic lymph nodes, or at least one large metastatic lymph. We looked to compare the data, and the first aspect was we looked into rare germline variants. So these rare germline missense mutations are those that are present in a minor allele frequency less than 1% in the 1,000 genomes database. And we found that such rare germline missense mutations in BRCA2 and FAT1, the two genes, actually they were enriched in patients who had lymph node metastasis compared to those who did not have. And we also found that in patients, both were not simultaneously mutated. Either of these genes were mutated in at least 43 of the patients who had lymph node metastasis. So 93% of these mutations were predicted by multiple algorithms to be deleterious in nature. However, we did not see any clustering of these patients in a particular location in these two genes. So there are some mutations known as hotspots, which are like mutations which cluster in a particular location in the gene. So one of the well-known hotspot mutations are in the TP53 that are associated with cancer. So there are about 5 very well-known hotspots in TP53. We found that somatic mutations present in at least 5 patients occurred in only 3 hotspots: the R175H, R248Q or W, and R282Q or W. We found a significantly higher frequency of the lymph node positive patients had one of these hotspot mutations compared to those who did not have lymph node metastasis. So TP53 hotspot mutations were enriched in patients who had lymph node metastasis. There's the other gene called caspase-8, and that has a hotspot reported in the TCGA, that's the arginine 233 codon. And we found that this mutation again was enriched in lymph node positive patients compared to lymph node negative individuals. The next feature that we looked into was how are the mutations mapped in various pathways. So we focused on the DNA repair pathway genes. And there are about 145 DNA repair pathway genes, out of which we found non-silent somatic mutations or rare non-silent germline mutations were found in 74 of these 145 genes, which were enriched in lymph node positive patients compared to those who did not have lymph node metastasis. And these genes were enriched in the non-homologous end joining pathway, the NHEJ pathway, but not the other pathway repair pathway genes. When we looked into the somatic mutation data, we found that there are multiple pathways like cell adhesion molecules, Hedgehog signaling, and TCA cycle and mRNA surveillance pathways that were again significantly enriched in somatic single nucleotide variants and indels in lymph node positive patients. Protein-protein functional interaction networks was used to find genes mutated either in the germline or somatically to identify molecular pathways that were metastatic in nature, so that were involved in metastasis. And we found that the mitotic G2M phase pathways, the NHEJ pathway, homologous recombination pathway, the tight junction KEK pathway, and the fatty acid and the MHC class 2 pathway, antigen presentation pathways, were again statistically significantly enriched in lymph node positive patients. We also looked into chromosomal instability by looking into these looking into copy number data, and we found that there were specific evidences of chromosomal instability in homologous recombination pathways, like the BRCA2, RBBP8, and the other genes, and again, chromosomal instability in the mitotic G2M transition pathways that were significantly altered in lymph node positive patients. These mitotic G2M phase pathways genes are involved in centrosome maturation and separation of stage for the spindle assembly. 64% of the lymph node positive patients who had a somatic or rare germline mutation in the mitotic G2M transition pathway also had chromosomal instability. And these observations were again validated in the TCGA HNSCC copy number variation and RNA-seq data set. So in summary, we identified 5 genomic features which actually predict lymph node metastasis in OSCCGB. These were the somatic hotspot mutations in TP53 and caspase-8, rare germline mutations in BRCA2 and FAT1 genes, somatic and rare germline mutations in NHEJ pathway and mitotic G2M pathway, and chromosomal instability. When we undertook clustering of the unsupervised clustering using these data, we found that these 5 features were able to accurately classify 89.2% of the lymph node positive patients and 80% of the lymph node negative patients. And unsupervised clustering also, using these 5 features, we found these 2 clusters, out of which the cluster 1 consisted of 92% of the patients of cluster 1 were lymph node negative patients and 72% were lymph node positive. So we also found that interesting differences in the survival. BRCA2 mutated patients had BRCA2 mutated patients who had lymph node metastasis had lower disease-free survival. However, mutations in the NHEJ pathway in lymph node positive patients were associated with higher disease-free survival. We also looked into the mitochondria because historically it's associated with the glycolytic phenotype of the cancer cells, but now we know that the bioenergetic shift of tumor cells are not permanent, and they can be reprogrammed based on the tumor microenvironment conditions. We did whole genome sequencing of the mitochondrial genome, To cut a long story short, we found again that the signature of the mutations in the mitochondria was different from that of the nuclear genome. It was C2T in the context of TCN, which was the predominantly mutated compared to the tobacco signature. So the mitochondrial mutations were not driven by tobacco, but intrinsic processes in the cell, particularly exposure to reactive oxygen species. We found that the somatic mutations in mitochondria had evidence of positive selection because the DN by DS ratios of these mutations were in the different electron transport chain complexes were higher in more than one in the somatic mutation landscape. We also found that patients who harbored non-synonymous somatic mutations in the mitochondria had higher occurrences of lymph node metastasis compared to those who did not harbor any somatic mutation in the mitochondria. Looking further, deeper, we found that patients who had, again, this non-synonymous somatic mutations in the electron transport chain 3, 4, and 5 had a higher cases of frequency of lymph node metastasis compared to those who had mutations in the electron transport chain 1. So we had some ideas about how this can happen. Probably the 3, 4, and 5 mutations result in more ROS production, which inhibits PTEN, and that can go on and activate AKT. And we did see some expression changes downstream of AKT, which supports our hypothesis. Now, I again shift gears. We looked into the epigenomic dysregulations in OSCCGB. There's various reasons why looking at epigenomic changes in cancer are very important. Generally, promoter hypermethylation of the tumor suppressors is a feature, a very prominent feature in all tumor types, and that results in dysregulation of downstream signaling pathways. We looked into, we found that in predominantly the DNA methylation profiling was, when we found that the high proportion of loci that were hypermethylated were different in different chromosomes. The chromosome 19 had the highest proportion of hypermethylation. And when we looked down further into the data, we found that DNA methylation profiling identified about 4,861 protein coding genes, out of which significant differential methylation in the promoter of 2,318 genes were observed, out of which most of them were hypermethylated and about 700 genes were hypomethylated. We looked into, again, we looked into the expression of these genes, and we found that 2,207 protein coding genes were differentially methylated, and there were some 46% were upregulated and 54% were downregulated. Looking at the methylation data, when we did that again an unsupervised clustering, we found that there were two clusters. The larger cluster of patients who belong, patients who belong to the larger clusters, they had more cases of higher stage of tumors, T3, T4, or had higher occurrences of lymph node metastasis, were in N1 or N2. So this was one feature that was interesting, that the methylation data could be used to correlate with clinical phenotypes. Then we looked into genes that were so DNA methylation caused an downstream expression change. So the understanding is, if the promoter is hypermethylated, the expression will be downregulated. Promoter is hypomethylated, the expression will be upregulated. So we found that about 209 genes were found to have significant negative correlation between DNA methylation and expression values. And out of these, we found that epigenetic downregulation due to significant promoter methylation were present in about 156 genes. There were about 10 genes. These 10 genes had the highest, strongest negative correlation, out of which we know that and out of these genes that were identified, we found that many of these genes can be altered by known drugs. And we also looked into the pathways to which these genes, which were epigenetically dysregulated, belong to, and we found that PPAR signaling pathway was implicated. Interestingly, the arachidonic acid metabolism pathway, which was earlier we had found by the mutation data analysis, we found that that pathway is also implicated in the epigenomic and the expression data set. We also found that B-cell receptor signaling pathway was involved. And also, interestingly, genes, there was some upregulation, significant upregulation of PDL1, CD80 genes, and CD86, and CTLA4. So these all shows that there is so the epigenomic dysregulation causes a suppression of the effector T cell response, and PD1, PDL1 can be a potential target in OSCCGB. So I will end here with, the talk that I gave, the data that I presented, and the findings are findings of a large team that worked on the India project, which was led by Professor Partha Majumdar from NIBMG and Dr. Rajiv Sareen from Tata Actrec, and my colleague Nidhan Biswas from NIBMG, who is a very vibrant and a key driver of this data analysis and findings, and the India project teams at Actrec and NIBMG. We also have other collaborators who participated in some generation of some of these data sets. And we also, I'd like to thank Department of Biotechnology, Government of India, who funded the entire ICGC Indian participation. With this, I would like to end. We'd be happy to hear from you, and would like to invite you in better times to our institute. This is our genomics lab. It's a National Genomics Core now, and it's a factory that we churn out genomic data for various diseases, and this is our institute. And we have a very, very active and vibrant genome clock. Thank you very much.
Thank you very much, Arindam. That was a great talk, a lot of data and a lot of insights into the various processes that lead to buccal oral cancer. I see a question for you in the chat box. The question is do you see any instance of transposon mobilization in precipitation of oral cancer?
Although in our data set, we have not yet, we are looking into it. We have not come out with our findings so far, but if you consult our pan-cancer analysis of whole genomes paper of the ICGC and TCGA that was recently published in 2020, there were some cancer types where this feature has been found to be very dominant, even in some HNSCC tumor types.
Are there any questions from YouTube?
It's none. There is none.
Okay, thank you. So, Arindam, there is no other question for you, and thank you very much for a great talk. And please stand by. There may be more talks, and we'll reach out to you if there are more talks at the end and if we have time left. Our next speaker is Professor Andreas Reis. He is a clinician originally, and he worked in France, Germany, and Chile. Subsequently, he moved to the World Health Organization and took a great interest in promoting public health ethics. That's what he does in WHO. He is a co-lead of one of the major ethics groups that moves and not only moves, but also promotes policies or promulgates policies and guidelines that WHO publishes from time to time with respect to ethics in public health. I personally think that this is a very important talk because much of the kind of talks that especially Mark Daly and Arindam gave that involves collection of data and biospecimens from individuals it's important to understand what ethical issues are in public health, because oftentimes those kinds of collections, even with informed consent, etc., can lead to deterioration of ethics in public health. So we need to take appropriate safeguard, and that's the reason why Andreas is a leader in this particular area, such that there is no negative impact on the community resulting from ethical issues with respect to genomics research. Without further ado, Andreas, would you give your talk, which is the contribution of ethics to public health? Thank you very much for accepting our invitation. We look forward to your talk. Andreas.
Please unmute yourself.
Andreas,
please unmute yourself, please.
Okay, I'm sorry about this.
and full screen, please.
Yes.
Thank you.
So first, let me thank Professor Majumdar and also INSA for inviting me to this very important meeting. I'm very happy that I can be with you, at least through WebEx, if not in person. And I hope I can make an important input into your discussion. Of course, my time in the laboratory doing my MD thesis is about 20 years away, so I cannot claim I'm any expert in genomics or genetics, but I have listened with great interest to these fantastic presentations that preceded me. So the topic of my talk is very different from these other more technical presentations. It is really about the contribution that ethics can make to public health and the essential function, I would argue, that ethical analysis has for public health. Here's a short overview of what I will be talking about. After some background slides, I would like to go right into the key issues that are arising in public health ethics. I will speak briefly about global health ethics at the WHO, since this is the institution I'm working at, and then I will offer some conclusions and an outlook. Ethics in medicine has actually a very long tradition. Medical doctors especially have been concerned with ethical issues since the ancient times. And here you see that also this concern about ethical issues has not only been one in Europe. Most people will know the Hippocratic Oath, which you see on the left top, but actually around the world. So on the right top side, you will see a picture of Sun Tzu Miao, who already in 600 years AD wrote about medical ethics in China, and his most famous text is called On the Absolute Sincerity of Great Physicians. And this is sometimes called also the Chinese Hippocratic Oath. On the left bottom, you see a very important source from the Middle East, written by Avicenna or Ibn Sina on the medical regulations. And then finally, on the right bottom, you see the stela called the Code of Hammurabi, which is really one of the oldest and most significant writings in the history of humankind. And it deals in the 2nd millennium BC with a variety of daily rules, including a doctor's code of conduct. So really worldwide, thousands of years of ethical concerns in medicine. What is more recent is the discipline of research ethics, or ethics of research with human subjects. And although there have been some discussions going back to the 19th century, really the origins of this discipline go back to the Nuremberg doctors' trial, which occurred after the horrible atrocities that were committed in Nazi Germany in the concentration camps under the guise of research. And in 1947, the Nuremberg Code was formulated with a main article saying never should be any research be done on human subjects without their consent, and then several other rules. And then there were two more significant documents that followed the Declaration of Helsinki, which many of you will know, issued by the World Medical Association for the first time in 1964, and then the SIOMS guidelines from 1991. Both documents are regularly updated. Just to go on with the clarification of terminology, you also are all familiar with the term bioethics. This is a term that was first mentioned in the 1930s, but then again in the 1950s. It is quite a generic term because it comes from bios, which means life in Greek, and so basically describes all the ethical issues that arise in humans, animals, and plants. And most importantly, 4 basic ethical principles have been formulated. This is referred to often as the Georgetown mantra. And you see the principles here on the right: autonomy, beneficence, non-maleficence, and justice. And then finally, I come to the discipline of public health ethics, which is a relatively recent discipline originating around the turn of the millennium, around 2000. And it grew mostly out of moral philosophy and common law. And as in contrast to research ethics or bioethics or medical ethics, it has a very different focus, which is the public health activities and programs. On this slide, you will see a comparison of what medical ethics and public health ethics focuses on. So the traditional medical ethics focuses on the individual patients and also the doctor-patient relationship. And it explores and enunciates the duties of doctors and nurses. In contrast to this, public health ethics focuses on the health of populations and not individuals, on health programs and policy issues, on scientific evidence, public participation, and also on preventive measures. And here you see some of the main principles of public health ethics. I will not go through them in detail, but just to say that 2 of the conditions, the first 2 have to do with evidence and the evidence base. So only public health measures that are absolutely necessary and that are reasonable and effective to solve a problem are in line with ethics. And just to take a recent example of COVID-19, there have been lots of discussions whether masks or social distancing actually works. Then proportionality is another very important issue. So the discussion has been whether a lockdown is proportional, whether it's sort of out of proportion for the situation. So these discussions reflect that principle of proportionality. Then distributive justice and equity, I think that's self-explanatory. Trust and transparency, which is very important for public policy making and decision making. And finally, I want to mention the Syracuse principles, which are actually from human rights. So the public health measures have to be based on a legitimate objective. They should be the least restrictive and intrusive means, and they should not be arbitrary or unreasonable or even discriminatory. With this short background, I would like to come to more practical issues and the application of these public health ethics principles to key issues in public health ethics. The first issue I want to discuss is the infectious disease area and the pandemics. And here we see a few examples of publications on things like tuberculosis, HIV, vector-borne diseases, and such as malaria and dengue, and also influenza and pandemic influenza and outbreaks. This is an area that is really front and center in public health ethics because many ethical issues arise in the prevention, in the research, in the response to these epidemics and pandemics. Here, just one example of a recent WHO document on ethics and outbreaks. And I'm showing that to illustrate some of the ethical issues that arise. So, for example, what are the obligations of governments and the international community? How to allocate scarce resources? Can you restrict the freedom of movement? How to do proper research during outbreaks? What to do with all these sort of miracle cures and offers or unproven interventions? How should they be regulated? The storage of biological specimen and also the sharing of specimen internationally, and ethical issues in deploying foreign human healthcare workers, and just to name a few. Of course, I think that's all on our mind, and so I couldn't help bringing in the COVID-19 pandemic, although I think some of us are also becoming a bit wary of always discussing this, but it is at the moment at the center of attention also for the field of public health ethics. And WHO has established several ethics working groups around these topics. So one important area is really the research for the COVID-19 vaccines and therapeutics. And we're at this moment about to publish a paper on the continued use of placebo arms in vaccine trials, which is a very thorny but important issue. I think I already mentioned on the previous slide that there are always lots of ethical debates about restrictive measures, physical distancing, and basically taking away the freedom to move around from people during pandemics. There's a big question, again, as in Ebola, about the fair allocation of scarce medical resources. And I think you're all probably following with interest how the vaccines will be rolled out. And this is a question both at an international level, but also within a country, what would be the first priority groups? And most countries would probably favor healthcare workers, frontline workers, and give priority to vulnerable populations. But there's lots of discussion, both internationally and in many countries, how to handle this. And similar discussions have been had for the allocation of ventilators, for example. Then there are lots of issues about public health surveillance and tracking of movement, tracking of people, of diseases, and how to protect all the related data that's gathered. And finally, an issue that always comes up, in how far do healthcare workers have a duty to sacrifice their health and life in the interest of their patients? And in relation to this question that has recently come up, should there be a mandatory vaccination, not for the population generally, but maybe for doctors or for people working in senior citizens' homes? Is this an ethical claim to make? And that's also an important point of discussion at the moment. And of course, there are numerous other ethical questions related to COVID-19 that we're addressing at this time. I briefly mentioned already the area of public health surveillance. This is a critical field for public health, sometimes called the eyes and ears of public health. And it raises multiple ethical issues. So, for example, is there informed consent required for surveillance activities? Or if you discover diseases or sick people during surveillance activities, in how far are you required ethically to provide the treatment, or what is the standard of care that needs to be provided, and WHO issued guidance in this very important area a few years ago. Another very important issue, and this was actually the #1 priority of WHO before COVID-19 started, and it continues, of course, to be one of the key focus areas, is universal health coverage, and the question of how we can reach everyone in need with the best available services. And here is a quote from our DG Tedros, who said, "For me, the key question of universal health coverage is an ethical one. Do we want our fellow citizens to die because they are poor, or millions of families impoverished by catastrophic health expenditures because they lack financial risk protection?" So ethics is really at the heart, and is the motivation for WHO to promote universal health coverage in all countries. And there are many questions in relation to that. We already talked about the question, in the face of scarcity, who will get priority access? Who will make these decisions? What kind of bodies would it be? Just the Ministry of Health, or a national ethics committee, or a national allocation committee, and so forth? How can we ensure that the limited resources are actually reaching the people? What would be a fair process for making these decisions? And then a question related to social determinants. We can have the best services in a clinic if then the people are treated and then go back to their poor housing conditions, etc. That is, of course, also not serving the purpose. So all these services have to be integrated with a drive to improve the social and environmental determinants of health. Another very big issue for public health ethics is the issue of organ tissue and cell transplantation and trafficking. And this is an area where WHO has already issued several guidelines and guiding principles, and is continually revising these principles about every 10 years. There's a question about coercion and exploitation of vulnerable donors or sellers to richer people. There is a question of consent for donors, whether it would be by default, opt out, or it would be opt in. And there's also a big issue, because there are long waiting lists on equitable access to organs that does not discriminate. Another key area which is gaining importance is the area of health promotion and non-communicable diseases. This really has to do with the question, how far should the government and should the state interfere with individual behavior, and how much coercion, if any, should a government use to promote healthy behavior? And one example is so-called sin taxes that would tax sugar or maybe trans fats to discourage people from buying unhealthy foods. But of course, this has also to do with alcohol prevention or smoking prevention. And on the left top, you see, for example, these plain packaging of cigarettes in Australia. And some people will argue this goes too far. This is sort of over the top, and is not in line with individual freedom. Others, and certainly WHO, will argue that this is very much necessary and needed for public health campaigns for the greater good of discouraging smoking. My final topic I will talk about in terms of key areas is the ethics of new technologies. And this is really something that has been gaining a lot of importance, not only in the world, but also at WHO. So the first topic here is human genome editing. And I don't have to tell you about CRISPR-Cas9. You know far more than I would about this topic. The fact is that there are many ethical concerns that arise with human genome editing. We also are acutely aware that global solutions are needed. It's not sufficient if just one country will agree on a government's framework. This is why over a year ago, WHO already established an international expert panel to discuss the ethical issues and governance options and advise the Director General on how to move forward and how to promote transparent and trustworthy practices in this area. The working group has been meeting frequently, and a report is expected in early 2021. Oh, sorry, I skipped one slide. The other application of CRISPR-Cas9 is, of course, for gene drives in mosquitoes. And there is a big potential for improving vector control and disease control in this area. At the same time, there are some risks attached to this. And WHO has just come out with a statement on gene drives in mosquitoes a couple of weeks ago. Some of the issues are how to best weigh the risk and benefits. The community acceptance of genetically modified mosquitoes is a big question. Some experts will say there could be potential harms for biodiversity. And of course, there's always the potential for international spread, as vectors don't know borders. And so this requires really a cross-border collaboration and international governance. The other big new technology, I think that's really on our mind, is big data and artificial intelligence. And as you know, all the medical journals, but also the public journals are full of news every day, maybe almost as much as COVID, with new applications, new possibilities of artificial intelligence and how it can in the future revolutionize medicine and public health. This is why WHO about a year ago established an expert group of about 20 experts from various disciplines, and we have been extremely fortunate that we have Professor Majumdar as one of the co-chairs in this working group, and he and the other co-chair, Effie Baena, have really been guiding this group very well towards the publication of a WHO guidance document, which will be published towards the end of the first quarter in next year. Some of the ethical issues that arise here. The first one is really quite basic. Given that in some of the lower and middle income countries, even the cheapest interventions cannot be afforded by everyone, how cost effective and sustainable are AI tools, in particular in these countries? So that's a very basic decision-making point for Ministry of Health, which also has, of course, ethical ramifications for accessing and universal health coverage for other services. The second one is how can we ensure data privacy, especially across borders? And then another very important one is the doctor-patient or nurse-patient relationship, which is really, I would say, characterized normally by a human interaction. How would this change if robots and AI will take over some of the services. Then we have seen that there's quite some danger of AI leading to bias and discrimination. This has to do with the database that the data that are fed into the algorithm. And we've seen, for example, that some machines have been trained to recognize melanoma, skin cancer in white people, and then they could not well recognize melanoma in black people because of the biased databases that was used to train. And another question is the digital divide, which is already existing. And the question is really whether AI will exacerbate that or will actually remedy it. And then there are two issues in relation to accountability and responsibility for decision making. If an AI algorithm is used, who is responsible if there are any mistakes? Is it the developer? Is it the hospital? Is it the doctor that remains responsible ultimately for these clinical services? These are very complicated questions. And finally, there is an important issue because of the great role that private industry is playing in this field. And there is quite a lot of challenges with the commercialization of healthcare using AI. One recent document that is sort of at the intersection of COVID-19 and digital health is about ethics of contact tracing or proximity tracking. And I know that in India also there are various apps of this kind. And it's really about surveillance, about data protection, and other issues which are extremely important. And now there's a discussion about how to replace the WHO yellow vaccination card that probably all of you know by sort of an app or a digital passport. And there are lots of ethical issues with this as well, of course. At the end of my talk, let me just say 2-3 words about global health ethics at WHO. I think I've already highlighted some of the documents and publications along my presentation, but just a bit more information about our work. So WHO has, a year ago, the member states have decided on a global program of work. And this is really quite a strategic framework for WHO for the next couple of years. And as you will see, health ethics is really at the heart of this global program of work, and actually articulating ethical and evidence-based policy options is one of WHO's 6 core functions. The document says WHO must ensure that policymakers and implementers keep ethics at the heart of their decision-making. And the program of work also gives particular importance to ethics of new and emerging scientific disciplines, such as genomics, epigenetics, gene editing, artificial intelligence, and big data, and universal health coverage. But you see the 2 big areas are new technologies and UHC. Just to say that WHO is not only developing guidance documents such as on genome editing or artificial intelligence or outbreaks, but we are also implementing them in countries, and especially with the help of our regional offices, we are doing quite a lot of trainings normally, which of course now are all online because of the situation. We also are greatly aided by our global network of collaborating centers for bioethics. One of them is actually a very recent one, is situated in Bangalore in India. and it complements greatly our network of 12 centers. Finally, I would like to offer some conclusions and an outlook. I have talked about some of the major concerns of ethics in public health, but of course there are many other topics such as antimicrobial resistance, dementia, and informed consent issues, personalized medicine that was already mentioned for different reasons, climate change, biobanking, and many others. So this was really not exhaustive. But I hope I have shown you that public health ethics is an essential component of public health. And it is important because it will strengthen the accountability of decision-making and the actions. It will also facilitate the trust and the compliance of people in public health measures if they see that it's in line, it's all done in line with the international ethical standards. I hope I have also demonstrated that most of the key issues in global health ethics cannot be solved at a national level alone. So for many of these issues, notably the pandemics such as COVID, but also things like genome editing or AI, there needs to be a global consensus and global solutions that then can be implemented or must be implemented at the national level. And with this, I would like to thank you very much for your attention.
Thank you, Dr. Rice, for that very nice presentation, starting with the Nuremberg trials until big data and artificial intelligence, genome editing, and so on. So this is the end of our symposium. Essentially, one of the reasons why I thought that this last talk was very important is because as we are unraveling the power of genomics on impact on human health and disease, there is a lot of automation and big data that are being generated. And unless we take proper precaution, ethical precaution of using this big data, it can have a negative impact on human health at the community level. And so we need to be cognizant, and we need to be, we need to take care of the fact that we wouldn't use much of this data in any inappropriate ways, so that it has a negative impact on human society and human communities. So to that an effect, it was very nice to hear Dr. Andres Reyes to describe to us the various aspects of ethics in medical genetics. One of the questions that has come up, Andreas, is could you comment on ethics of placebo arm for drug trials in debilitating diseases with no treatment?
You have to unmute yourself. You have to unmute yourself.
Andreas, you need to unmute yourself. Did you hear the question?
So my mouse was stuck. Yeah. Thank you for your warm words, Partha. Yeah. So the ethics of placebo use has been one of the thorniest questions in research ethics for the last decades. Even Salk, when he tested the first vaccines, was discussing this issue. And then in HIV in 2000, there was again this question in prevention of mother-to-child transmission trials in Africa, whether placebo could be used. There are various guidelines I mentioned. So the SIOMS guidelines, but also the Declaration of Helsinki guidelines spell out the conditions under which placebo can be used. And basically, it is permitted only in exceptions. And this is especially when there is no gold standard, when there's no standard treatment available. Then you can make a case for using exceptionally placebo. So if there are no treatments available in these diseases that you're mentioning, then it would be in line with ethics. We'll not have to look at the detail, but it would be in line with ethics to use placebo arms in these trials. Over.
Thank you, Andreas. Are there any questions for Andreas on YouTube?
No, there is none, but there are a lot of words of appreciation for the program and work of Professor Majumdar and his group, as well as the speeches.
Thank you. I don't know, Mark Stoneking, are you still there? There is a question that's been repeated a couple of times. So if you are still online.
I'm still here.
Hi, Mark. So, Mark, one of the questions is you talked about the Bashia Kars cave in Tibet. So the question is, did the Denisovans have sex with the Tibetans?
Well, it seems like the Denisovans had sex with the ancestors of all East Asians and Oceanians, since we see a signature of Denisovan ancestry now widespread across East Asia at very low levels, but it is detectable. It seems to be about background levels. I don't know of any study that's shown higher levels overall of Denisovan ancestry in Tibet, so I suspect that they have roughly the same levels of as all other groups. But for this one haplotype that confers high altitude adaptation, of course, they have a much higher frequency of this than any other population does.
Thanks, Mark. I think there are no other questions. So let me just wrap up this symposium before I hand over the microphone to Professor Singhvi and Professor Chandrima Saha. I think the symposium covered a wide range, starting from our evolutionary ancestries and histories and its impact on human health and disease, to actually what we have found using those evolutionary methods and so on in order to pinpoint genomic underpinnings of human disease. And then we moved on to talk about some specific human diseases such as IBD, autism, schizophrenia, and oral cancer. Finally, the ethics of all of this, the ethical impact of all of this kind of genomics research on human health and disease was discussed in the last talk by Dr. Andreas Reis. I wish to thank all of the speakers for their time and for their insightful talks, and most importantly, for their consent to give these wonderful talks. I also thank the Indian National Science Academy, in particular the president, Professor Chandrima Saha, and Professor Ashok Singhvi, for calling on me to organize this. And I think we all enjoyed the 4 wonderful talks. Thank you very much. I hand it over to Professor Chandrima Saha.
I will.
I think Professor Singhvi has to come.
Yes. I humbly thank Professor Majumdar and all the 4 speakers. Ever since I met Professor Majumdar, I found him a mesmerizing person, and it's no wonder that he was able to collect 4 mesmerizing speakers, taking the whole range of arcing genomes to the future of ethical and medical practices. I think he was so mesmerizing that even the internet was well behaved today, and there was not a single disturbance for the first time I saw it. So there is some kind of a magic that we have with you, Partha. I also like to compliment that India has joined major consortia, and the work in the cancer was very impressive. We are grateful to Professor Rice, because he talked about the issues of ethics, which are should be uppermost in our mind. And it could not be at a time more appropriate than this, because we are now struggling with various issues, COVID to artificial intelligence to future of medicine. We hope we will see you again sometimes in INSA. Mark, you have been wonderful, because you essentially traced out the history of where we came from and how much of messing up we have been doing. I hope it stops somewhere soon. And Mark Daly essentially shared the current status of genomic medicine. One was worried that we don't have, as of now, it looks very complex, but I'm sure in the years to come, they will sort it out in some detail. But it's a complex subject. And I was extremely proud to hear Dr. Arindam because he essentially told how much Indians are working on the cancer front and other areas. I wish he had more time to talk to us because, and hope we can arrange another talk for him because there's an enormous amount of data he showed, which we need time to assimilate. I warmly thank all the speakers, and I request all of you to give a big clap to for them in front of your screen. We can't do it in person, but a clap. And we hope that we'll see you, all of you, sometimes in person in India. And you'll be welcome to the Indian National Science Academy anytime you came to India. Thank you, Partho. Thank you, all the speakers. And give a big hand to all of you. So with this, we conclude the symposium today. We'll meet tomorrow again at 4 p.m. with a symposium on cybersecurity and issues, which will also have a legal expert speaking to us. And then we'll have a symposium on actually women in science. And I can tell you it will be a very powerful symposium. So I urge you to join. If you don't join, it will be at your peril. Thank you so much. Have a nice time and stay safe. Thank you, Partha.
Thank you. Thank you.
Thank you very much. Thank you very much. Good night.
Thank you, Partha.
Good morning, good afternoon.
Thank you, Partha.